Hello, and welcome. It's good that you're here. If you've arrived feeling tired, or restless, or simply in need of something gentle to accompany you as the day fades, then you've come to the right place. There's no need to do anything at all right now. You don't need to remember everything, or take notes, or even stay awake. You can simply let yourself settle in, wherever you are, and allow these words to wash over you like a quiet tide. Tonight, we're going to explore something truly remarkable, something happening inside you at this very moment. Even as you breathe and blink and let your shoulders drop, we're going to talk about the most relaxing facts surrounding cellular recycling processes. These are the hidden, tireless systems that keep your body clean, renewed, and humming along in the background of your life. They work without fanfare, without your conscious effort, like a devoted night crew that tidies up while you sleep. And there's something deeply comforting in knowing that even when you rest, your cells are caring for you. If you enjoy this kind of gentle science, you're welcome to follow along for more. But there's no pressure. Right now, all that matters is that you're comfortable, that you're breathing slowly, and that you're ready to drift into the microscopic world where renewal happens in whispers. So let's begin. Deep within every cell of your body, there's a process unfolding that resembles nothing so much as a careful, continuous housekeeping. It's called autophagy, a word that comes from the Greek for self-eating. Though that sounds far harsher than the reality, autophagy is gentle. It is your cell's way of recycling their own parts, breaking down what is worn out or damaged, and turning it into something useful again. It happens all the time, quietly, without you noticing, and it keeps you whole. Imagine a room in your home that slowly accumulates clutter over time. Papers pile up, old containers gather dust, things you once needed but no longer use begin to crowd the corners. Autophagy is like a patient hand that comes through, sorts what can be saved, discards what cannot, and leaves the space clear and ready. Your cells do this constantly. They recognize when a protein has become misfolded, when a damaged organelle is no longer functioning, when something inside has simply worn out from the work of living. And instead of letting it sit there, they wrap it up, take it apart, and reuse the pieces. This process was discovered relatively recently, though it has been happening since the first cells emerged billions of years ago. In 2016, a scientist named Yoshinori Ohsumi won the Nobel Prize for his work uncovering the mechanisms of autophagy. He spent years watching yeast cells under a microscope, observing how they survive starvation by eating parts of themselves. It was elegant, it was efficient, and it revealed a truth that applies to nearly every living thing. Life is not just about building up, but also about breaking down, recycling, and beginning again. When you fast, even for a short time, autophagy increases. Your cells sense that nutrients are scarce, and they respond by becoming more diligent recyclers. They turn inward, scanning for anything that can be dismantled and converted into energy or raw materials. Old mitochondria, damaged membranes, clumps of misfolded proteins. All of these become resources. It's a kind of molecular thrift, a wisdom born from billions of years of survival. And it happens without you having to think about it, without you having to will it into being. Your body knows. There is something profoundly restful in that knowledge. You do not have to manage every tiny process. You do not have to remind your cells to clean themselves, to repair what's broken, to recycle what's no longer needed. It happens automatically, like breathing, like the beating of your heart. You are being cared for from the inside out in ways you will never fully see. Let's move a little deeper into the structures that make this recycling possible. Inside each of your cells, there are small compartments called lysosomes. They are sometimes called the stomachs of the cell, though that's a simplification. Lysosomes are tiny sacs filled with enzymes, specialized proteins that can break down almost anything: carbohydrates, proteins, lipids, even entire organelles. They are powerful, but they are also carefully controlled. The enzymes inside a lysosome are kept separate from the rest of the cell by a membrane, a thin barrier that ensures they only do their work when and where it's needed. When autophagy occurs, the cell forms a double membrane structure called an autophagosome. This autophagosome surrounds the damaged or unneeded material, enclosing it like a soft bubble. Then, it travels through the cell until it finds the lysosome. The two membranes fuse, and the contents are delivered into the lysosome's interior, where the enzymes get to work. Slowly, methodically, they dismantle the material into its basic components: amino acids, fatty acids, sugars. These components are then released back into the cell, where they can be reused to build new proteins, generate energy, or support other vital functions. It's a cycle. Nothing is truly wasted. What was once a worn-out part becomes the foundation for something new. And this cycle happens thousands of times a day, in trillions of cells, throughout your entire body. It is one of the most fundamental rhythms of life, and it asks nothing of you but to exist. Lysosomes themselves are fascinating. They were discovered in the 1950s by a Belgian scientist named Christian de Duve, who was studying insulin and stumbled upon these mysterious sacs filled with digestive enzymes. At first, he wasn't sure what they were for, but over time, it became clear that lysosomes were essential for cellular digestion and waste management. Without them, cells would become clogged with debris, unable to function, eventually dying under the weight of their own accumulated damage. In some genetic disorders, lysosomes don't work properly. The enzymes are missing or malformed, and waste begins to build up inside cells. These conditions, known as lysosomal storage diseases, can be devastating, but they also highlight just how crucial this recycling system is. When it works invisibly, seamlessly. When it fails, the consequences ripple outward. It's a reminder that health is often about the small things, the hidden processes, the quiet maintenance that goes on beneath the surface. But tonight, your lysosomes are working. They're breaking down what needs to be broken down, clearing space, making room for renewal. And you don't have to do a thing. Let's consider the mitochondria, those small, bean-shaped organelles often called the powerhouses of the cell. They generate the energy your cells need to function, converting nutrients into a molecule called ADP, which fuels nearly every process in your body. But mitochondria are also constantly under stress. They produce reactive oxygen species as a byproduct of energy production, and over time, this can damage their own membranes and DNA. Left unchecked, damaged mitochondria can become harmful, releasing signals that trigger inflammation or even cell death. So your cells have a solution: mitophagy. This is a specialized form of autophagy that targets mitochondria specifically. When a mitochondrion becomes too damaged to repair, the cell tags it for removal. An autophagosome forms around it, just as it would around any other piece of cellular debris, and delivers it to a lysosome. There, the mitochondrion is dismantled, its components recycled. In its place, the cell can generate new, healthy mitochondria, ensuring that energy production continues smoothly. Mitophagy is quality control. It's the cell's way of ensuring that only the best, most functional mitochondria remain. And it happens constantly, especially in tissues that demand a lot of energy, like your muscles, your heart, and your brain. These organs rely on healthy mitochondria to keep going, and mitophagy is one of the reasons they can sustain their activity over a lifetime. There's something almost tender about this process. Your cells are not just machines. They are systems capable of self-assessment, self-correction, and self-renewal. They notice when something isn't right, and they take steps to fix it. They protect you, even from themselves. As you lie here, your cells are performing mitophagy. Old mitochondria are being gently dismantled. New ones are being made. Energy is flowing, quietly, and continuously, keeping you warm, keeping your heart beating, keeping your thoughts drifting through the soft space between waking and sleep. Now let's turn our attention to proteins, the workhorses of the cell. Proteins do almost everything. They catalyze chemical reactions, transport molecules, provide structure, send signals, defend against invaders. But proteins are also fragile. They are long chains of amino acids folded into precise three-dimensional shapes. And those shapes are essential to their function. If a protein misfolds, it can lose its ability to do its job. Worse, misfolded proteins can clump together, forming aggregates that interfere with cellular processes. Your cells have several systems for dealing with misfolded proteins. One of the most important is the ubiquitin proteasome system. When a protein is damaged or misfolded, it gets tagged with a small molecule called ubiquitin. This tag is like a flag, a signal that says this protein needs to go. The tagged protein is then delivered to a proteasome, a large, barrel-shaped complex that unfolds the protein and breaks it down into individual amino acids. These amino acids are then released back into the cell, where they can be used to make new, properly folded proteins. The proteasome is remarkably efficient. It can degrade a protein in just a few seconds, and it's highly selective, targeting only those proteins that have been tagged with ubiquitin. This ensures that healthy, functional proteins are left alone, while damaged ones are swiftly removed. But not all misfolded proteins are handled by the proteasome. Some are too large, or too tangled, or clustered into aggregates that the proteasome can't process. That's where autophagy steps in again. Autophagosomes can engulf large protein aggregates and deliver them to lysosomes, where they're broken down by enzymes. This is especially important in neurons, which are long-lived cells that accumulate a lot of protein damage over time. Autophagy helps keep neurons clean, preventing the buildup of toxic aggregates that can lead to neurodegenerative diseases. In fact, many neurodegenerative conditions, including Alzheimer's and Parkinson's disease, are associated with failures in protein quality control. When autophagy and the proteasome system can't keep up with the accumulation of misfolded proteins, those proteins begin to form plaques and tangles that disrupt brain function. It's a sobering reminder of how important these recycling processes are. But it's also a reminder that, for most of your life, they work beautifully. They keep your neurons clear, your synapses firing, your thoughts flowing. Right now, as you rest, your cells are folding proteins correctly, tagging the ones that aren't, and quietly removing them before they can cause harm. It's a kind of invisible diligence, a background hum of care that never stops. Let's think about the endoplasmic reticulum, or ER, a vast network of membranes inside your cells where many proteins are made and folded. The ER is like a factory floor, bustling with activity. Ribosomes attach to its surface and churn out new proteins, which are then threaded into the ER's interior, where they fold into their proper shapes. But sometimes, things go wrong. A protein might misfold, or the ER might become overloaded with too many proteins at once. When this happens, the cell experiences what's called ER stress. To cope with ER stress, cells activate a response called the unfolded protein response, or UPR. The UPR has several strategies. First, it slows down protein production, giving the ER time to catch up. Second, it ramps up the production of chaperone proteins, which help other proteins fold correctly. Third, it activates autophagy and the proteasome system to clear out misfolded proteins. And if the stress is too severe, if the cell can't recover, the UPR can trigger a controlled form of cell death called apoptosis, sacrificing the cell to protect the organism as a whole. The UPR is a beautiful example of cellular resilience. It's a system that senses imbalance and responds with a coordinated effort to restore order, and it relies heavily on recycling. By breaking down misfolded proteins and reusing their amino acids, the cell can reduce the burden on the ER and give itself a chance to recover. This process is happening in you right now. Your cells are constantly monitoring the state of their adjusting protein production, deploying chaperones, clearing out mistakes. It's a dance of feedback and correction, and it keeps your cells healthy even in the face of stress. There's a certain poetry in the idea that your cells are always listening to themselves, always adjusting, always striving for balance. They are not rigid or static. They are fluid, responsive, alive in the truest sense. Let's consider the concept of cellular senescence. As cells age, some of them stop dividing and enter a state called senescence. Senescent cells are still alive, but they no longer contribute to tissue growth or repair. Instead, they secrete a mix of inflammatory molecules, growth factors, and enzymes that can affect neighboring cells. This is sometimes called the senescence-associated secretory phenotype, or SASP. For a long time, scientists thought senescent cells were simply a byproduct of aging, a kind of cellular retirement with no real purpose. But more recent research suggests they play a role in wound healing, tissue remodeling, and even tumor suppression. However, when senescent cells accumulate in large numbers, they can contribute to chronic inflammation and age-related diseases. The body has ways of dealing with senescent cells. One of the most important is the immune system, which can recognize and clear senescent cells through a process that involves autophagy and other recycling mechanisms. Immune cells engulf senescent cells and break them down, much like they would with pathogens or cellular debris. This helps keep tissues clean and functional. There's also growing interest in therapies that target senescent cells, called senolytics. These drugs are designed to selectively eliminate senescent cells, potentially slowing aging and reducing the risk of age-related diseases. It's a frontier of research that hinges on our understanding of cellular recycling and waste management. But even without intervention, your body is already working to manage senescent cells. Your immune system is patrolling, identifying cells that have outlived their usefulness and clearing them away. It's another layer of care, another form of recycling, another way your body looks after itself. Let's drift now to the idea of cellular turnover. Your body is not a fixed structure. It is constantly renewing itself, replacing old cells with new ones. Some tissues turn over rapidly. The lining of your gut is replaced every few days. Your skin cells are renewed every few weeks. Your red blood cells are replaced every few months. Other tissues, like bone and muscle, turn over more slowly. And some cells, like most neurons, last a lifetime. But even in long-lived cells, individual components are constantly being recycled. Proteins are broken down and rebuilt. Membranes are remodeled. Organelles are replaced. This means that even if a cell itself is old, its parts are often quite young. It's a bit like the ship of Theseus, the ancient philosophical puzzle. If you replace every plank of a ship over time, is it still the same ship? In your body, the answer is yes. You remain you, even as your cells recycle themselves piece by piece. This turnover is essential for health. It allows your body to adapt to changing conditions, to repair damage, to clear out toxins and waste. And it all depends on recycling. Without autophagy, without lysosomes, without the proteasome, cellular turnover would grind to a halt. Cells would become clogged with debris, unable to renew themselves, and tissues would begin to fail. But that's not happening. Right now, your cells are turning over. Old parts are being dismantled. New parts are being made. You are, in a very real sense, constantly being reborn, one molecule at a time. There's a deep comfort in this. You're not static. You're not fixed in place. You are a process, a flow, a continuous act of renewal. And that renewal is happening even now, even as you rest, even as you do nothing at all. Let's think about the role of fasting and caloric restriction in cellular recycling. When you eat less or when you go without food for a period of time, your cells shift into a different metabolic state. Insulin levels drop, glucagon and other hormones rise, and autophagy increases. Your cells, sensing that nutrients are scarce, become more efficient recyclers. They break down more of their own components, extracting every bit of usable material, and use it to sustain themselves. This is why fasting has been linked to various health benefits, from improved metabolic health to increased longevity. It's not magic. It's simply that fasting activates pathways that promote cellular cleaning and repair. Autophagy clears out damaged organelles and misfolded proteins, reducing the burden of cellular waste. This, in turn, can reduce inflammation, improve mitochondrial function, and enhance overall cellular health. Of course, fasting isn't necessary for autophagy to occur. It happens all the time, even when you're well-fed. But fasting can give it a boost, a chance to work more intensively. It's like giving your cells a spring cleaning, a deeper sweep than the daily tidying they usually do. And there's something peaceful about the idea that sometimes doing less, eating less, striving less, demanding less of yourself can be a form of care. It can give your cells the space they need to focus on maintenance, on repair, on renewal. It's a reminder that rest is not laziness. Rest is essential. Let's consider the lysosomes' role in nutrient sensing. Lysosomes are not just passive recycling bins. They are also signaling hubs, capable of detecting the nutritional state of the cell and communicating that information to the rest of the cell. On the surface of lysosomes, there are protein complexes that sense the availability of amino acids. When amino acids are abundant, these complexes activate a signaling pathway called mTOR, which promotes cell growth and protein synthesis. When amino acids are scarce, mTOR is inhibited and autophagy is activated. This means that lysosomes are at the center of a critical decision: grow or recycle. When nutrients are plentiful, the cell grows, builds new structures, divides. When nutrients are limited, the cell recycles, conserves, cleans itself. It's a balance, a constant negotiation between building up and breaking down, and the lysosome is the mediator. This role makes the lysosome far more than just a digestive organelle. It's a sensor, a communicator, a decision maker. It integrates information about the cell's environment and adjusts cellular behavior accordingly. And it does this quietly, without fanfare, as part of the seamless coordination that keeps you alive. Right now, your lysosomes are sensing, signaling, adjusting. They are helping your cells decide how to allocate resources, how to balance growth and maintenance, how to stay healthy in a changing world. And you don't have to think about it. It just happens. Let's turn to the idea of cellular stress and resilience. Life is full of stresses, both external and internal. Your cells are exposed to toxins, to oxidative damage, to temperature fluctuations, to mechanical forces. They experience stress from overwork, from lack of nutrients, from infections. But they are also remarkably resilient. They have evolved a suite of responses that allow them to cope with stress, to adapt, and to survive. Autophagy is one of the most important stress responses. When a cell is stressed, autophagy ramps up, clearing out damaged components and providing the cell with the resources it needs to repair itself. This is true for many types of stress: oxidative stress, nutrient deprivation, hypoxia, infection. Autophagy is a kind of cellular first aid, a way of stabilizing the situation and buying time for recovery. But autophagy is not just reactive, it's also preventive. By continuously clearing out damaged organelles and misfolded proteins, autophagy reduces the baseline level of cellular stress. It keeps the cell clean and functional, preventing small problems from becoming big ones. It's maintenance, not just repair. This dual role, both preventive and reactive, makes autophagy essential for long-term health. It's why cells that can't perform autophagy are more vulnerable to stress and more likely to die. And it's why boosting autophagy through fasting, exercise, or other interventions can have such broad health benefits. Your cells are resilient. They are built to withstand stress, to adapt, to recover. And much of that resilience comes from their ability to recycle, to renew, to start fresh. It's a strength that runs deep, woven into the very fabric of life. Let's think about the relationship between autophagy and aging. As we age, autophagy tends to decline. Cells become less efficient at recycling their components, and damaged organelles and misfolded proteins begin to accumulate. This accumulation contributes to many of the hallmarks of aging: mitochondrial dysfunction, cellular senescence, inflammation, loss of proteostasis. In other words, a decline in autophagy is not just a symptom of aging, it's one of the causes. But the relationship goes both ways. Enhancing autophagy can slow some aspects of aging. In laboratory animals, interventions that boost autophagy, such as caloric restriction, fasting, or treatment with drugs like rapamycin, have been shown to extend lifespan and improve health span, the period of life spent in good health. The animals live longer, and they remain healthier for more of their lives. This doesn't mean that autophagy is a fountain of youth. Aging is complex, influenced by many factors, and no single intervention can stop it. But it does suggest that maintaining robust autophagy is an important part of healthy aging. It's one of the ways we can support our cells as they carry us through the years. And there's something hopeful in that. Aging is inevitable, but how we age is not entirely fixed. The choices we make, how we eat, how we move, how we rest, can influence the health of our cells, the efficiency of our recycling systems, the quality of our later years. We have some agency, even in the face of time. Right now, your cells are performing autophagy. They are doing what they can to keep you healthy, to slow the accumulation of damage, to give you as many good years as possible. They are working for you, even as you rest. Let's consider the role of exercise in promoting autophagy. When you exercise, especially when you engage in aerobic or endurance exercise, your muscles experience a kind of controlled stress. They use up energy. They generate reactive oxygen species. They sustain small amounts of damage. In response, your cells activate autophagy to clear out damaged mitochondria and proteins, and to support the repair and remodeling of muscle tissue. Over time, regular exercise enhances the capacity for autophagy. Your cells become better at recycling, more efficient at clearing waste, more resilient to stress. This is one of the reasons exercise has such broad health benefits. It's not just about building muscle or improving cardiovascular fitness. It's also about maintaining the health of your cells at a fundamental level. And the effects of exercise-induced autophagy extend beyond muscle. Exercise promotes autophagy in the brain, where it helps clear out protein aggregates and supports cognitive function. It promotes autophagy in the liver, in the heart, and fat tissue. It's a systemic effect, a whole body boost to cellular health. But you don't have to be an athlete to benefit. Even moderate exercise, walking, gentle cycling, stretching, can stimulate autophagy. It's about movement, about challenging your body just enough to activate its maintenance systems. And there's something lovely about the idea that taking care of yourself can be as simple as taking a walk, as gentle as moving your body through space. Your cells respond to kindness. They respond to care. And exercise, in its many forms, is a way of caring for them. Let's think about the connection between autophagy and the immune system. Your immune cells rely heavily on autophagy to function. When a macrophage, a type of immune cell, engulfs a bacterium, it uses autophagy-related machinery to deliver that bacterium to a lysosome, where it's destroyed. This process, called xenophagy, is a form of autophagy that targets foreign invaders. Autophagy also helps immune cells manage their own health. Immune cells are highly active, producing large amounts of proteins and reactive molecules as they fight infections. This activity generates a lot of cellular stress, and autophagy helps clear out the damage, keeping immune cells functional and ready to respond. In addition, autophagy plays a role in presenting antigens, the molecular flags that help the immune system recognize and remember pathogens. By breaking down proteins and displaying fragments of them on the cell surface, autophagy contributes to the adaptive immune response, the system that gives you long-lasting immunity after an infection or vaccination. When autophagy is impaired, immune function suffers. Cells are less able to clear infections, more prone to inflammation, and less effective at mounting adaptive responses. This is why autophagy is considered a critical part of immune health, a behind-the-scenes process that supports your body's defenses. Right now, your immune cells are using autophagy to patrol your body, to clear away pathogens, to manage their own stress. They are protecting you quietly and continuously in ways you will never directly perceive. And that protection depends on recycling, on the ability to break down and renew. Let's drift toward the idea of cellular communication and autophagy. Cells don't exist in isolation. They are part of tissues, organs, systems. They communicate with each other through chemical signals, through physical contact, through shared environments. And autophagy is part of that communication. When a cell undergoes autophagy, it can release signals that affect neighboring cells. For example, dying cells that are being cleared by autophagy can release molecules that attract immune cells, promoting tissue repair and remodeling. Senescent cells, as we mentioned earlier, secrete factors that influence the behavior of surrounding cells. And even healthy cells can release small vesicles, called exosomes, that contain proteins, RNA, and other molecules derived from autophagy. These signals create a kind of cellular conversation, a network of information that helps coordinate the behavior of tissues and organs. Autophagy is not just an internal housekeeping process. It's also a way for cells to communicate their state, to call for help, to contribute to the collective health of the body. This perspective shifts how we think about recycling. It's not just about cleaning up. It's about connection, about community, about the ways individual cells work together to create something larger than themselves. Your body is not a collection of isolated units. It's a symphony, and autophagy is one of the instruments. As you rest, your cells are communicating. They are sending and receiving signals, coordinating their activities, supporting each other. You are a community, a living network, and that network is taking care of itself. Let's think about the origins of autophagy deep in evolutionary time. Autophagy is ancient. It's found in nearly all eukaryotic organisms, from yeast to plants to animals. This suggests that it evolved early, perhaps more than 1 billion years ago, and the common ancestor of all complex cells. And it has been preserved, refined, and elaborated over countless generations because it is essential. Why is autophagy so fundamental? Because life is messy. Cells accumulate damage. Proteins misfold. Organelles wear out. And without a way to clear that damage, cells would quickly become dysfunctional. Autophagy is the solution that evolution found, a way to keep cells clean and functional over time. It's a form of cellular hygiene that makes long-term survival possible. In single-celled organisms, autophagy allows survival during starvation. A yeast cell, facing a lack of nutrients, can digest parts of itself to stay alive until conditions improve. In multicellular organisms, autophagy takes on additional roles: development, immune defense, tissue remodeling, aging. But the core function remains the same: recycling for survival. There's something humbling about this deep history. The autophagy happening in your cells right now is the same process that kept your ancestors alive, that allowed the first complex cells to thrive, that has been passed down through billions of years of life. You are connected to that history, part of an unbroken chain that stretches back to the very beginning. And there's comfort in continuity. You are not alone. You are part of something vast, something ancient, something that has endured. And as we drift deeper into this microscopic world of renewal, we find ourselves drawn to the elegant machinery of the proteasome, a structure so beautifully designed that it seems almost intentional in its grace. The proteasome is a barrel-shaped complex, a tiny cylinder that sits in the cytoplasm of your cells, waiting patiently for damaged or misfolded proteins to arrive. When a protein has outlived its usefulness or has become tangled in ways that make it dangerous, the cell marks it with a small tag, a molecule called ubiquitin. This tagging process is gentle, deliberate, like placing a soft label on something that needs attention. The ubiquitin molecules attach in chains, and these chains serve as signals, quiet whispers that guide the protein toward the proteasome's opening. Once inside, the protein is carefully unfolded and broken down into its component amino acids, which can then be reused to build new proteins. Nothing is wasted. Everything is recycled with precision and care. The proteasome works constantly, processing thousands of proteins every hour, and yet it does so without fanfare, without strain. It is one of the most active recycling systems in your body, operating in every cell, in every tissue, at every moment. When you sleep, the proteasome continues its work. When you wake, it is still there, quietly maintaining the integrity of your cellular machinery. This system is so essential that without it, cells would quickly become overwhelmed by damaged proteins, leading to dysfunction and disease. But with it, with this tireless molecular recycler, your cells remain clean, functional, and ready to respond to whatever the day brings. The proteasome represents a kind of quiet diligence, a form of cellular housekeeping that requires no conscious effort on your part, no thought, no intention. It simply happens, as naturally as breathing. And beyond the proteasome, there exists another layer of recycling, one that operates at the level of entire cellular compartments. When organelles become damaged or redundant, the cell does not simply discard them haphazardly. Instead, it employs a process called selective autophagy, in which specific organelles are targeted for degradation. We have already spoken of mitophagy, the recycling of mitochondria, but there are many other forms of selective autophagy, each with its own name and purpose. Hexophagy, for instance, is the recycling of peroxisomes, small organelles involved in breaking down fatty acids and detoxifying certain chemicals. When peroxisomes become old or damaged, they are enveloped by autophagosomes and delivered to lysosomes for breakdown. This ensures that the cell's metabolic functions remain efficient and that toxic byproducts do not accumulate. There is also ribophagy, the selective degradation of ribosomes, the molecular machines responsible for synthesizing proteins. Ribosomes are incredibly abundant in cells, especially in those that produce large amounts of protein, such as liver cells or immune cells. But when nutrients are scarce, or when certain ribosomes become defective, the cell can selectively recycle them, breaking them down into their nucleotide and amino acid components. This provides the cell with raw materials during times of stress and prevents the buildup of non-functional ribosomes that could interfere with protein synthesis. Ribophagy is particularly active during starvation, acting as a kind of emergency resource recovery system. It is a testament to the cell's ability to adapt, to prioritize, to make the most of what it has. Another fascinating form of selective autophagy is reticulophagy, the recycling of portions of the endoplasmic reticulum. The ER is a vast network of membranes that extends throughout the cell, and it is constantly being remodeled in response to changing needs. When parts of the ER become damaged or are no longer needed, they are selectively removed through reticulophagy. This process helps maintain the ER's structure and function, ensuring that protein folding and lipid synthesis can continue without interruption. The ER is so central to cellular health that its careful maintenance is essential, and reticulophagy is one of the key mechanisms by which this maintenance is achieved. It is a quiet, continuous process, one that reflects the cell's commitment to balance and order. And then there is nucleophagy, a rare and intriguing form of autophagy in which portions of the nucleus itself are recycled. The nucleus, the command center of the cell, is generally well protected and stable, but under certain conditions, such as extreme stress or nutrient deprivation, small pieces of the nuclear envelope or nuclear contents can be selectively degraded. This is not a common process, and it is still being studied, but it reveals the extent to which cells are willing to sacrifice even their most critical structures in order to survive. Nucleophagy is a reminder that cellular recycling is not just about routine maintenance, but also about survival, about making difficult choices when resources are scarce. It is a process that speaks to the resilience and adaptability of life at the smallest scales. All of these forms of selective autophagy are regulated by specific proteins and signaling pathways, each tailored to recognize and target particular organelles or structures. This specificity is crucial because it allows the cell to recycle only what needs to be recycled, preserving what is still functional and useful. The cell does not act blindly or randomly. It assesses, it evaluates, it makes decisions based on a constant flow of information from its internal environment. This decision-making process is remarkably sophisticated, involving multiple layers of regulation and feedback. It is a system that has been refined over billions of years of evolution, honed to perfection through countless generations of trial and error. And yet, despite its complexity, it operates smoothly, seamlessly, without conscious oversight. As we continue our journey through the landscape of cellular recycling, we come upon the concept of chaperone-mediated autophagy, or CMA, a process that is distinct from the autophagy we have discussed so far. In CMA, individual proteins are targeted for degradation without the need for an autophagosome. Instead, these proteins are recognized by chaperone proteins, molecular assistants that guide them directly to the lysosome. The targeted proteins contain a specific amino acid sequence, a kind of molecular address, that marks them for CMA. The chaperone binds to this sequence and escorts the protein to the lysosomal membrane, where it is unfolded and transported directly into the lysosome's interior. There, it is broken down by the lysosomal enzymes, and its components are released back into the cell for reuse. CMA is a highly selective process, more so than macroautophagy, because it targets individual proteins rather than entire portions of cytoplasm. It is particularly important during times of stress, such as prolonged fasting or oxidative damage, when the cell needs to quickly remove damaged or unnecessary proteins. CMA also plays a role in regulating the levels of certain key proteins, helping to maintain cellular homeostasis. For example, it can degrade transcription factors, enzymes, or structural proteins that are no longer needed, preventing them from interfering with cellular function. This precision makes CMA an essential component of the cell's quality control systems, a finely tuned mechanism that operates alongside other forms of autophagy to keep the cellular environment clean and balanced. The discovery of CMA was a significant milestone in our understanding of cellular recycling. It revealed that cells have multiple pathways for degrading proteins, each suited to different circumstances and different types of cargo. This redundancy is important because it provides the cell with flexibility and resilience. If one pathway is compromised, others can compensate, ensuring that essential recycling processes continue. This is a common theme in cellular biology: the presence of backup systems, alternative routes, and overlapping functions. It is a design philosophy that prioritizes robustness and reliability, and it is one of the reasons why cells are so remarkably durable and adaptable. And as we think about the many forms of autophagy, we begin to see a larger pattern, a theme of transformation and renewal that extends beyond individual cells to entire tissues and organisms. The processes we have been exploring are not isolated events. They are part of a continuous cycle of breakdown and synthesis, of death and rebirth, that sustains life at every level. This cycle is ancient, dating back to the earliest cells, and it is universal, present in every living organism from bacteria to humans. It is a fundamental principle of biology, a truth that underlies all of life's complexity and diversity. And it is a deeply reassuring truth because it tells us that renewal is always possible, that even in the face of damage and decay, there is a mechanism for restoration and healing. Let us now turn our attention to the role of cellular recycling and development, in the process by which a single fertilized egg becomes a complex, multicellular organism. During development, cells must undergo dramatic transformations, changing their shape, their function, and their identity. Autophagy plays a crucial role in these transformations. For example, during the metamorphosis of a tadpole into a frog, the tadpole's tail is reabsorbed through a process that involves extensive autophagy. The cells of the tail break down their own components, recycling the materials to support the growth of new structures, such as legs and lungs. This is a striking example of how autophagy can drive large-scale changes in body structure, how it can enable one form of life to give way to another. Similarly, in mammals, autophagy is essential during the early stages of embryonic development. Shortly after fertilization, the embryo undergoes a period of rapid cell division, but it has not yet established a connection to the mother's blood supply. During this time, the cells rely heavily on autophagy to generate the energy and building blocks they need to survive and grow. They recycle their own cytoplasm, breaking down proteins, lipids, and other molecules to fuel the process of cell division. This self-sustaining strategy allows the embryo to develop even in the absence of external nutrients, a remarkable feat of biological resourcefulness. It is a reminder that autophagy is not just a maintenance process, but also a creative one, enabling growth and transformation. Autophagy also plays a role in the elimination of structures that are no longer needed during development. For example, during the formation of fingers and toes, the cells in the tissue between the digits undergo programmed cell death, a process called apoptosis. Autophagy works alongside apoptosis to ensure that these cells are completely removed, preventing the formation of webbed hands or feet. This collaboration between autophagy and apoptosis is a common theme in development, as the two processes often work together to sculpt the body, removing excess tissue and refining structures. It is a process that requires precision and coordination, and it is guided by complex signaling pathways that respond to the body's developmental program. And beyond development, autophagy continues to play a role throughout life, supporting the ongoing maintenance and repair of tissues. In the adult body, many tissues undergo continuous turnover, with old cells being replaced by new ones. The lining of the intestine, for example, is replaced every few days, and the skin is replaced every few weeks. This constant renewal requires a steady supply of building blocks, and autophagy helps provide them by recycling old or damaged cellular components. In this way, autophagy supports the body's regenerative capacity, its ability to heal wounds, to replace worn-out tissues, and to adapt to changing conditions. It is a process that operates quietly in the background, unnoticed but essential, a foundation upon which the body's health and vitality rest. Let us now consider the role of cellular recycling in the nervous system, a tissue that is particularly vulnerable to the accumulation of damaged proteins and organelles. Neurons are long-lived cells, often persisting for the entire lifespan of an organism, and they are highly active, consuming large amounts of energy and producing large amounts of waste. Because neurons rarely divide, they cannot dilute their damaged components through cell division, as other cell types can. Instead, they rely heavily on autophagy and other recycling processes to maintain their health. In neurons, autophagy is particularly important for clearing away aggregated proteins, which can form toxic clumps that interfere with cellular function. These protein aggregates are a hallmark of many neurodegenerative diseases, including Alzheimer's, Parkinson's, and Huntington's disease. In a healthy neuron, autophagy acts as a protective mechanism, preventing the buildup of these aggregates and maintaining the neuron's ability to transmit signals. But when autophagy is impaired, either through aging, genetic mutations, or environmental factors, protein aggregates can accumulate, leading to neuronal dysfunction and death. This is why researchers are so interested in finding ways to enhance autophagy in the brain as a potential strategy for preventing or treating neurodegenerative diseases. Some studies have shown that interventions such as caloric restriction, exercise, and certain drugs can boost autophagy in neurons, reducing the burden of protein aggregates and improving neuronal health. These findings are still preliminary, but they offer hope that by supporting the brain's natural recycling processes, we might be able to slow or even reverse the progression of some of these devastating diseases. Autophagy in neurons is also important for maintaining the health of synapses, the connections between neurons where signals are transmitted. Synapses are dynamic structures, constantly being formed, strengthened, weakened, or eliminated in response to experience and learning. Autophagy helps regulate this process by removing old or damaged synaptic components, making room for new ones. This synaptic remodeling is essential for learning and memory, and it is a process that continues throughout life. Every time you learn something new, every time you form a new memory, your neurons are undergoing subtle changes at the synaptic level, and autophagy is there quietly supporting this process, ensuring that the machinery of learning remains functional and efficient. And as we think about the brain, we are reminded of the profound connection between cellular health and mental well-being. The processes we've been exploring, these quiet, invisible acts of recycling and renewal, are not abstract or distant. They are happening in your brain right now. As you listen to these words, as you process this information, as you drift towards sleep, they are part of what makes thought possible, what makes consciousness possible, what makes you you. And there is something deeply comforting in this realization, in knowing that even as you rest, even as you let go of the day's worries and concerns, your cells are working to maintain your health, to repair damage, to prepare you for tomorrow. You do not need to do anything. You do not need to try. You simply need to rest and let your body do what it does so well. Let us now explore the role of cellular recycling in the immune system, a network of cells and tissues that defends the body against infection and disease. Immune cells are among the most active cells in the body, constantly surveying for threats, responding to infections, and producing antibodies and other defensive molecules. This high level of activity generates a significant amount of cellular waste, and autophagy plays a crucial role in managing this waste. In immune cells, autophagy helps clear away pathogens that have been engulfed by the cell, a process called xenophagy. When a macrophage or other immune cell engulfs a bacterium or virus, the pathogen is enclosed in a membrane-bound compartment called a phagosome. Autophagy machinery can then target this phagosome, fusing it with a lysosome to create a phagolysosome, where the pathogen is destroyed by digestive enzymes. Xenophagy is a critical defense mechanism, helping to eliminate intracellular pathogens that might otherwise evade the immune system. Some bacteria and viruses have evolved strategies to escape from phagosomes and replicate within the host cell, but autophagy can detect these escapees and target them for destruction. This makes autophagy an important component of the cell's intrinsic immune defenses, a kind of backup system that operates alongside other immune mechanisms. In this way, cellular recycling is not just about maintaining cellular health, but also about protecting the body from external threats. It is a dual-purpose system, one that serves both housekeeping and defensive functions. Autophagy also plays a role in regulating the immune response, ensuring that it is neither too weak nor too strong. After an infection has been cleared, the immune system must return to a resting state, and autophagy helps facilitate this transition by removing excess immune molecules and signaling proteins. This prevents chronic inflammation, a condition in which the immune system remains activated even in the absence of a threat, causing damage to the body's own tissues. Chronic inflammation is associated with many diseases, including arthritis, cardiovascular disease, and cancer, and autophagy's role in resolving inflammation is an area of active research. By promoting the clearance of inflammatory signals, autophagy helps restore balance to the immune system, allowing it to respond effectively to new threats without causing unnecessary harm. In addition to its role in immune cells, autophagy also influences the function of the thymus, an organ that is central to the development of T cells, a type of white blood cell that is essential for adaptive immunity. The thymus undergoes a process called involution as we age, shrinking in size and producing fewer T cells. This decline in thymic function is one of the reasons why the immune system becomes less effective with age. Recent research has shown that autophagy is important for maintaining the health of thymic epithelial cells, the cells that support T cell development. Enhancing autophagy in these cells may help preserve thymic function and support immune health in older individuals. This is another example of how cellular recycling processes have far-reaching effects, influencing not just individual cells, but entire organ systems and overall health. And as we continue to explore the many roles of cellular recycling, we find ourselves drawn to the concept of cellular communication, the ways in which cells send and receive signals from one another. This communication is essential for coordinating the activities of different cells and tissues, ensuring that the body functions as a coherent whole. Autophagy plays a surprising role in this communication, particularly through the release of extracellular vesicles, tiny membrane-bound packages that cells use to exchange information. When a cell undergoes autophagy, some of the recycled materials are not fully degraded, but are instead packaged in vesicles and released into the extracellular space. These vesicles can be taken up by neighboring cells, delivering their contents and influencing the recipient cell's behavior. This process, sometimes called secretory autophagy, is a relatively new area of research, but it is already revealing fascinating insights into how cells communicate and coordinate their activities. For example, cancer cells can use secretory autophagy to release signals that promote tumor growth and suppress the immune response. On the other hand, healthy cells can use this process to share beneficial molecules, such as antioxidants or signaling proteins, with their neighbors. This form of cellular communication adds another layer of complexity to our understanding of autophagy, showing that it is not just an internal process, but also a social one, a way for cells to interact and support one another. It is a reminder that even at the microscopic level, life is fundamentally interconnected, with each cell influencing and being influenced by those around it. Let us now turn our attention to the role of cellular recycling in the aging process. a topic that has captured the imagination of scientists and the public alike. Aging is a complex phenomenon, influenced by many factors, including genetic, environmental, and lifestyle factors. But at the cellular level, aging is characterized by a gradual decline in the efficiency of maintenance and repair processes, including autophagy. As we age, the activity of autophagy tends to decrease, leading to the accumulation of damaged proteins, dysfunctional organelles, and other cellular debris. This accumulation contributes to many of the hallmarks of aging, including inflammation, oxidative stress, and cellular senescence. In essence, the decline in autophagy is both a cause and a consequence of aging, a vicious cycle that accelerates the aging process. But the good news is that autophagy is not fixed or immutable. It can be influenced by lifestyle factors, and there is growing evidence that enhancing autophagy can slow the aging process and extend health span, the period of life spent in good health. We have already mentioned caloric restriction and intermittent fasting as ways to boost autophagy, but there are other strategies as well. Regular physical exercise, for example, is a powerful inducer of autophagy, particularly in muscle and brain tissue. When you exercise, your cells experience a kind of beneficial stress, a signal that resources are being depleted and that recycling is needed. In response, autophagy is activated, clearing away damaged components and making room for new, healthy ones. This is one of the reasons why exercise is so beneficial for health and longevity, and why it is often described as a form of cellular rejuvenation. Certain dietary compounds have also been shown to enhance autophagy. Resveratrol, a compound found in red grapes and red wine, has been studied for its potential to activate autophagy and extend lifespan in model organisms. Curcumin, the active ingredient in turmeric, has similar effects. Green tea contains catechins, which can also promote autophagy. These compounds work by activating signaling pathways that regulate autophagy, such as the AMPK and SGRT1 pathways, which we discussed earlier. While the effects of these compounds in humans are still being studied, the preliminary evidence is promising, suggesting that dietary interventions could be a simple and accessible way to support cellular health and slow the aging process. There are also pharmaceutical approaches to enhancing autophagy. Rapamycin, a drug originally developed as immunosuppressant, has been shown to extend lifespan in mice and other model organisms by inhibiting mTOR and activating autophagy. While rapamycin is not yet widely used for anti-aging purposes in humans due to potential side effects, it has sparked interest in developing safer, more targeted drugs that can mimic its effects. Metformin, a common diabetes medication, also appears to have autophagy-enhancing effects and is being studied for its potential to promote healthy aging. These pharmacological approaches are still in the early stages of research, but they represent a promising frontier in the quest to understand and modulate the aging process. And as we think about aging and autophagy, we are reminded that the goal is not necessarily to live forever, but to live well, to maintain health and vitality for as long as possible. The processes we have been exploring, these quiet acts of cellular recycling and renewal, are not about achieving immortality. They are about supporting the body's natural capacity for repair and regeneration, about giving our cells the tools they need to function optimally. They're about quality of life, about remaining active, engaged, and healthy as we age. And this, perhaps, is the most reassuring aspect of cellular recycling. It is not a fantasy or a distant promise, but a real, tangible process that is happening in your body right now, and that you can support through simple, everyday choices. Let us now consider the role of cellular recycling in the context of sleep, the very state you may be entering as you listen to these words. Sleep is a time of profound restoration, a period when the body and brain engage in essential maintenance and repair processes. and autophagy is central to this restoration. During sleep, the brain's glymphatic system becomes more active, clearing away metabolic waste products that have accumulated during waking hours. This system works in parallel with autophagy, which is also upregulated during sleep, particularly in neurons. Together, these processes help remove toxic proteins, such as beta amyloid and tau, which are associated with Alzheimer's disease. This is one of the reasons why sleep is so important for brain health, and why chronic sleep deprivation is associated with an increased risk of neurodegenerative diseases. Autophagy during sleep is not limited to the brain. It occurs throughout the body, in muscle tissue, in the liver, in the immune system, and in every other organ and tissue. Sleep is a time when the body shifts from a state of activity and energy expenditure to a state of rest and repair. Hormonal changes that occur during sleep, such as the release of growth hormone, help promote autophagy and other restorative processes. In this way, sleep and cellular recycling are intimately connected, each supporting the other in a harmonious cycle of renewal. When you sleep well, you support your cell's ability to recycle and repair. And when your cells are healthy and functioning optimally, you sleep better. It is a virtuous cycle, one that underscores the importance of prioritizing rest and recovery in our busy lives. And as we drift further into this exploration, we find ourselves contemplating the elegance of cellular recycling, the way it embodies principles of efficiency, sustainability, and balance. In a world that often feels wasteful and chaotic, there is something deeply comforting in knowing that at the cellular level, nothing is wasted, everything is reused, and balance is constantly maintained. The processes we have been exploring are not just biological mechanisms, but also metaphors for how we might live our own lives. With care, with intention, with a commitment to renewal and letting go of what no longer serves us. They remind us that transformation is possible, that even the most damaged or worn-out parts of ourselves can be broken down and remade into something new and useful. Let us now explore the role of cellular recycling in the context of cancer, a disease that arises when cells lose their normal regulatory controls and begin to divide uncontrollably. Autophagy has a complex and somewhat paradoxical relationship with cancer. In healthy cells, autophagy acts as a tumor suppressor, preventing the accumulation of damaged proteins and organelles that could lead to mutations and malignant transformation. By maintaining cellular health and genomic stability, autophagy helps prevent the initial development of cancer. Many tumor suppressor genes, such as PTN and TSC1 and 2, work by promoting autophagy, and loss of these genes is associated with an increased risk of cancer. In this sense, autophagy is protective, a guardian against the forces that drive cancer development. However, once a cancer has developed, autophagy can play a different role. Cancer cells often experience high levels of stress due to rapid growth, limited nutrient supply, and the hostile environment of the tumor. In response to this stress, cancer cells can upregulate autophagy, using it as a survival mechanism to generate energy and building blocks. Autophagy allows cancer cells to adapt to harsh conditions, to survive chemotherapy and radiation, and to resist the immune system. In this context, autophagy is pro-tumorigenic, supporting the survival and growth of cancer cells. This dual role of autophagy in cancer has important implications for cancer treatment. In some cases, inhibiting autophagy might make cancer cells more vulnerable to therapy, while in other cases, enhancing autophagy might help prevent cancer from developing in the first place. Researchers are actively exploring ways to target autophagy in cancer treatment, developing drugs that can either inhibit or enhance autophagy, depending on the context. Chloroquine and hydroxychloroquine, drugs originally used to treat malaria, are being studied as autophagy inhibitors in cancer therapy. These drugs work by preventing the fusion of autophagosomes with lysosomes, thereby blocking the completion of autophagy. Clinical trials are underway to determine whether combining these drugs with conventional cancer treatments can improve outcomes for patients. On the other hand, some researchers are exploring whether enhancing autophagy in precancerous cells might help prevent the development of cancer. This is a rapidly evolving field, and our understanding of how to best modulate autophagy in cancer is still developing. The relationship between autophagy and cancer highlights the complexity of cellular recycling processes and the need for precision and context in any therapeutic approach. It reminds us that biological systems are not simple or one-dimensional, but are instead characterized by nuance, balance, and context dependence. What is beneficial in one situation may be harmful in another, and understanding these nuances is essential for developing effective treatments. This complexity is not a weakness, but a strength, a reflection of the adaptability and resilience of living systems. And it is a reminder that even as we seek to understand and manipulate these processes, we must do so with humility and care, recognizing that we are still learning, still discovering, still uncovering the mysteries of life at the cellular level. Let us now turn our attention to the role of cellular recycling in the context of the microbiome, the vast community of microorganisms that live in and on our bodies. The microbiome plays a crucial role in health, influencing digestion, immunity, metabolism, and even brain function. And emerging research suggests that the microbiome can also influence autophagy in host cells. Certain gut bacteria produce metabolites, such as short-chain fatty acids, that can modulate autophagy in intestinal cells and other tissues. These metabolites can activate signaling pathways that promote autophagy, helping to maintain the health of the gut lining and support immune function. In this way, the microbiome acts as a kind of external regulator of cellular recycling, influencing processes that are normally thought of as purely internal. The relationship between the microbiome and autophagy is bidirectional. Just as the microbiome can influence autophagy, autophagy can also influence the microbiome. Autophagy in intestinal cells helps regulate the immune response to gut bacteria, preventing excessive inflammation while still allowing the immune system to respond to harmful pathogens. Autophagy also plays a role in the secretion of antimicrobial peptides, which help shape the composition of the microbiome by selectively inhibiting certain bacteria. This interplay between autophagy and the microbiome is an area of active research, and it is revealing new insights into how the body maintains a healthy relationship with its microbial inhabitants. It is another example of the interconnectedness of biological systems, the way that processes at the cellular level are influenced by and influence the larger ecosystem of the body. And as we think about the microbiome and autophagy, we are reminded of the importance of a holistic approach to health, one that considers not just individual cells or organs, but the body as a whole, including the trillions of microorganisms that call it home. The processes we have been exploring, these quiet acts of cellular recycling, do not occur in isolation. They are part of a larger web of interactions, a complex network of feedback loops and signaling pathways that connect every part of the body. Understanding this network and how to support it through lifestyle choices is key to maintaining health and preventing disease. And the good news is that many of the same interventions that support autophagy, such as a healthy diet, regular exercise, and adequate sleep, also support a healthy microbiome. These are not separate goals, but complementary ones, different facets of the same underlying commitment to health and well-being. Let us now explore the role of cellular recycling in the context of the cardiovascular system, the network of heart and blood vessels that delivers oxygen and nutrients to every cell in the body. The cardiovascular system is under constant stress, subjected to the mechanical forces of blood flow, the oxidative stress of high metabolic activity, and the inflammatory signals of the immune system. Autophagy plays a crucial role in maintaining the health of cardiovascular cells, including cardiomyocytes, heart muscle cells, and endothelial cells, the cells that line blood vessels. In cardiomyocytes, autophagy helps remove damaged mitochondria and protein aggregates, maintaining the cell's ability to contract and generate force. This is particularly important because cardiomyocytes are long-lived cells that rarely divide, and they rely heavily on autophagy to maintain their health over the lifespan. In endothelial cells, autophagy helps protect against oxidative stress and inflammation, two major contributors to cardiovascular disease. When endothelial cells are healthy, they produce nitric oxide, a molecule that helps relax blood vessels and maintain normal blood pressure. But when endothelial cells are damaged, they produce less nitric oxide and more inflammatory signals, contributing to the development of atherosclerosis, the buildup of plaques in the arteries. Autophagy helps prevent this damage by removing oxidized lipids and damaged proteins, supporting the endothelium's protective functions. Studies have shown that enhancing autophagy in endothelial cells can improve vascular health and reduce the risk of cardiovascular disease. This is another example of how cellular recycling processes have far-reaching effects, influencing not just individual cells, but entire organ systems and overall health. The heart itself undergoes significant changes throughout life, and autophagy plays a role in these changes. During fetal development, the heart relies on autophagy to support its rapid growth and differentiation. In the adult heart, autophagy helps maintain cardiac function in the face of stress, such as hypertension or ischemia, reduce blood flow. And in the aging heart, autophagy helps mitigate the effects of accumulated damage, supporting cardiac health and function. However, as with other tissues, autophagy in the heart tends to decline with age, contributing to the increased risk of heart disease in older individuals. Finding ways to support autophagy in the heart through lifestyle interventions or pharmacological approaches is an important goal of cardiovascular research. And as we think about the cardiovascular system, we are reminded of the importance of movement and activity in supporting cellular health. When you exercise, your heart rate increases, your blood vessels dilate, and your muscles demand more oxygen and nutrients. This increased activity generates a kind of beneficial stress, a signal that resources are being used and that recycling is needed. In response, autophagy is activated in cardiovascular cells, clearing away damaged components and making room for new, healthy ones. This is one of the reasons why regular physical activity is so beneficial for cardiovascular health, and why sedentary lifestyles are associated with an increased risk of heart disease. Movement is not just good for your muscles and your lungs, it is good for your cells, supporting the very processes that keep them healthy and functional. The body's quiet work of renewal extends into territories you might not expect, reaching into the very architecture of how cells remember, respond, and rebuild themselves across the span of a lifetime. There is something deeply comforting in knowing that even as you rest, even as your conscious mind drifts towards sleep, your cells are engaged in an ancient conversation with themselves, a dialogue written in molecules and membranes, spoken in the soft language of recycling and renewal. Consider for a moment the endoplasmic reticulum, that vast network of folded membranes we touched upon earlier. When this structure becomes damaged or overwhelmed, the cell doesn't simply abandon it. Instead, a specialized form of autophagy called autophagy quietly disassembles the excess or damaged portions, recovering the building blocks and maintaining just the right amount of this essential organelle. It's a process of careful editing, like a gardener pruning a plant not to harm it, but to help it flourish. The cell knows its own needs with a wisdom that requires no thought, no planning, only the patient execution of patterns refined across billions of years. And there are receptors on the ER itself, small proteins that serve as tags, gentle signals that say this portion here, this fold of membrane, has served its purpose and may now be recycled. These receptors are recognized by the autophagy machinery with the same tender specificity we've seen elsewhere, ensuring that only what needs to go is taken, only what has completed its service is returned to the pool of raw materials. Nothing is wasted, nothing is lost. Everything continues in a new form. The lipid droplets in your cells, those small spheres of stored fat that serve as energy reserves, are also subject to this watchful recycling. When energy is needed, a process called lipophagy selectively targets these droplets, wrapping them in autophagosomal membranes and delivering them to lysosomes where the fats are broken down into fatty acids your mitochondria can burn for fuel. It's a remarkably efficient system, one that ensures your cells can access stored energy precisely when it's required, maintaining a steady supply even during times of scarcity. The process is so finely tuned that it responds to your body's energy state moment by moment, increasing when you fast or exercise, quieting when nutrients are abundant. This recycling of lipids has profound implications for metabolic health. When lipophagy functions well, cells avoid the toxic accumulation of excess fat, maintaining their sensitivity to insulin and their capacity to respond appropriately to energy signals. When it falters, lipid droplets can accumulate inappropriately, contributing to metabolic dysfunction. But the beauty of a system is that it can be supported, gently encouraged through the same practices that enhance autophagy in general. Periodic fasting, regular movement, adequate rest. Your lifestyle whispers to your lipophagy machinery, and it listens. Beyond the recycling of large structures, there exists an even more intimate form of quality control, one that operates at the level of individual molecules moving through cellular membranes. The ERAD system, which stands for endoplasmic reticulum-associated degradation, identifies misfolded proteins before they even leave the ER, pulling them back out through the membrane and delivering them directly to proteasomes for destruction. It's a checkpoint system, a gentle but firm gatekeeper that ensures only properly formed proteins continue their journey into the cell. The misfolded ones are not punished or discarded carelessly. They are simply and quietly recycled, their amino acids returned to the pool, ready to be tried again. This quality control extends to the Golgi apparatus as well, that elegant stacking of membranes where proteins are further modified and sorted. Autophagy, the selective autophagy of Golgi components, maintains the health and appropriate size of this organelle, removing damaged sections and allowing the cell to rebuild them as needed. The Golgi, like so many cellular structures, is not a static monument, but a living, breathing, constantly renewed architecture. What appears solid and permanent under the microscope is actually a river, a flow of molecules being continuously replaced, refreshed, reborn. Even the nucleus, that central chamber where your DNA resides, participates in selective autophagy. Nucleophagy can target specific nuclear components, including portions of the nuclear envelope and even fragments of chromatin under certain conditions. This allows the cell to remodel its nuclear architecture, adjusting the very structure that houses its genetic information in response to developmental signals or stress. It's a remarkable degree of plasticity, a willingness to reshape even the most central and protected compartment of the cell when circumstances require it. The cytoskeleton, that intricate scaffold of protein filaments that gives your cells their shape and allows them to move, is also subject to renewal through autophagy. Damaged or excess microtubules, actin filaments, and intermediate filaments can be selectively degraded, preventing the accumulation of dysfunctional structural elements that might impair cell movement or division. This process is particularly important in neurons, where the cytoskeleton must maintain highways for transport across extraordinarily long distances, from your spinal cord to your toes, a journey that in some neurons spans more than a meter. The recycling of cytoskeletal components ensures these highways remain clear and functional throughout your life. There's a beautiful circularity to how autophagy regulates itself. The very process of autophagy produces signals that can either enhance or limit further autophagy, creating feedback loops that keep the system balanced. When autophagy is active, it can degrade certain regulatory proteins that would otherwise inhibit it, allowing the process to continue. But it can also degrade proteins that activate autophagy, creating a natural break that prevents excessive self-digestion. The cell walks a middle path, neither too much nor too little, guided by an internal wisdom that has no need of conscious oversight. The connection between autophagy and circadian rhythms runs even deeper than we've discussed. Many of the key autophagy genes show circadian patterns of expression, meaning they're naturally more active at certain times of day. In most organisms, including humans, autophagy tends to peak during the night and early morning, during the fasting period when cellular resources need to be mobilized and cellular components need to be refreshed. This daily rhythm of breakdown and renewal is synchronized with your sleep-wake cycle, with your eating patterns, with the fundamental rotation of the Earth. Your cells are not separate from the cosmos. They dance to its rhythms. This circadian regulation of autophagy may be one reason why disrupted sleep patterns and irregular eating schedules can have such profound effects on health. When you eat late at night, you provide nutrients at a time when your cells are primed for breakdown and recycling, creating a conflict between opposing signals. When you sleep irregularly, you desynchronize the many clocks running in your cells, causing autophagy and other processes to fall out of their proper rhythm. But the system is forgiving, returning to regular patterns, even after periods of disruption, allows these rhythms to re-establish themselves, the cellular orchestra finding its tempo once again. The role of autophagy in the liver deserves special attention, for the liver is perhaps the body's most metabolically active organ, a chemical factory running thousands of reactions simultaneously. Hepatocytes, the main cells of the liver, rely heavily on autophagy to manage their enormous metabolic burden. They use it to break down glycogen stores when blood sugar drops, to degrade lipid droplets when fatty acids are needed, to remove damaged mitochondria that accumulate from the intense oxidative metabolism the liver performs. Liver autophagy is so important that its impairment is associated with virtually every form of liver disease, from fatty liver to hepatitis to cirrhosis. But when liver autophagy functions well, the liver demonstrates a remarkable capacity for regeneration. You can remove a large portion of a healthy liver, and the remaining tissue will grow back, hepatocytes dividing and expanding until the organ reaches its proper size again. Autophagy supports this regeneration, clearing away cellular debris, providing building blocks for new growth, maintaining quality control as cells divide rapidly. It's one of the most dramatic examples of regeneration in the adult human body, and autophagy is quietly essential to it. In the kidneys, autophagy protects the delicate filtering units, called nephrons, from the constant stress of processing blood and producing urine. Kidney cells are exposed to high concentrations of metabolic waste products, to fluctuations in salt and water, to the mechanical stress of fluid flow. Autophagy helps them manage this challenging environment, removing damaged components and maintaining the specialized structures required for filtration. As we age, kidney autophagy tends to decline, and this decline is associated with the gradual loss of kidney function that many people experience. Supporting autophagy may be one way to protect kidney health across the lifespan. The connection between autophagy and stem cells reveals another layer of this process's importance. Stem cells, Those remarkable cells that can divide to produce more stem cells or differentiate into specialized cell types rely on autophagy to maintain their unique properties. Autophagy keeps stem cells in a state of quiet readiness, clearing away damaged components that might trigger premature differentiation or senescence. When a stem cell does differentiate, autophagy often increases dramatically, helping to remodel the cell's interior to match its new identity. The process of becoming a muscle cell, a neuron, or a blood cell involves not just making new components, but also selectively removing old ones, sculpting the cell from within. Hematopoietic stem cells, which give rise to all your blood cells, provide a particularly clear example. These cells reside in your bone marrow in a low-oxygen environment, and they maintain high levels of autophagy to survive in these conditions and to preserve their stem cell properties. When they differentiate into red blood cells, there's a massive wave of autophagy that removes the nucleus and most organelles, leaving behind a cell that's essentially a bag of hemoglobin, optimized for carrying oxygen. This dramatic remodeling, this willingness to discard so much of the cellular machinery, is made possible by autophagy working at full capacity. In the skin, autophagy supports the constant renewal that keeps this barrier between you and the world intact and functional. Skin cells are born in the deep layers and gradually move outward, differentiating and flattening as they go, eventually dying and forming the tough, protective outer layer of dead cells that you shed continuously. Autophagy is active throughout this journey, helping cells manage the stress of differentiation and the programmed destruction of their internal components. It also helps skin cells respond to ultraviolet light, removing damaged proteins and DNA that accumulate with sun exposure. Supporting skin autophagy may be one mechanism behind the protective effects of certain skincare ingredients and practices. The process of wound healing also depends on autophagy in multiple ways. When you cut your skin or injure a tissue, cells at the wound edge must migrate into the damaged area, proliferate to fill the gap, and produce new extracellular matrix to knit the tissue back together. Autophagy provides energy for these demanding processes and also helps remodel the cells as they change from stationary tissue residents to mobile, active wound healers. Inflammatory cells that arrive to clean up debris and fight infection also rely on autophagy to manage the oxidative stress they generate and to process the material they engulf. A wound healing is a symphony of cellular activities, and autophagy is one of the quiet instruments playing throughout. Even in bone, that's seemingly solid and unchanging tissue, autophagy plays a vital role. Osteocytes, the cells embedded in the bone matrix, use autophagy to survive in their isolated, low-nutrient environment. These cells live for years or even decades, maintaining the bone around them and communicating with other bone cells through tiny channels. Autophagy helps them endure this long vigil, recycling components to sustain themselves and responding to mechanical stress by adjusting their activity. When you walk, run, or lift weights, you create forces that travel through your bones, and osteocytes sense these forces and respond by signaling to other cells to strengthen the bone. Autophagy supports this mechanosensing function, helping osteocytes maintain the sensors and signaling machinery they need. The interplay between autophagy and inflammation is intricate and bidirectional. Inflammation can trigger autophagy as cells respond to stress signals, and autophagy in turn can modulate inflammation, generally dampening excessive inflammatory responses. This is partly because autophagy removes damaged mitochondria that might otherwise release inflammatory signals, and partly because it degrades certain inflammatory signaling proteins directly. In chronic inflammatory conditions, from arthritis to inflammatory bowel disease, autophagy is often impaired, and this impairment may contribute to the persistent inflammation. Restoring autophagy function is being explored as a therapeutic strategy for these conditions. The gut, that long tube running through your body, is a particularly important site of autophagy activity. The intestinal epithelium, the layer of cells lining your gut, is one of the most rapidly renewing tissues in your body, with cells being replaced every few days. This rapid turnover requires enormous amounts of autophagy to manage the cellular remodeling involved. Gut cells also use autophagy to handle the unique stresses of their environment. Exposure to food antigens, bacterial products, digestive enzymes, and mechanical forces. Panneth cells, specialized gut cells that produce antimicrobial peptides, rely on autophagy to manage the stress of producing and secreting these powerful proteins. There's also a fascinating connection between gut autophagy and the microbiome. Some of the bacterial components that make it across the gut barrier can trigger autophagy in intestinal cells, and this autophagy can capture and degrade bacteria that have invaded the cells. It's a form of cellular immunity called xenophagy, which we touched on earlier, but in the gut it takes on special importance because of the sheer number of bacteria present just on the other side of the epithelial barrier. The conversation between your gut cells and your gut bacteria involves autophagy as one of its languages, a way for your cells to maintain boundaries while still benefiting from the presence of their microbial partners. Autophagy's role in cancer is complex and context-dependent in ways that reveal the deep ambiguity of biological processes. In healthy cells, autophagy can prevent cancer by removing damaged mitochondria and other components that might otherwise generate mutations or inflammatory signals that promote transformation. Many tumor suppressors, genes that prevent cancer, actually work in part by promoting autophagy. But once a cancer is formed, those same cancer cells often use autophagy to survive in the harsh environment of a tumor, where nutrients and oxygen are scarce. Autophagy becomes a tool for survival, allowing cancer cells to weather the storms of their own disordered growth. This has made autophagy both a potential target for cancer prevention and a potential target for cancer therapy, though in opposite directions. Enhancing autophagy in healthy individuals might reduce cancer risk. Inhibiting autophagy in established tumors might make them more vulnerable to treatment. Researchers are exploring both approaches, developing drugs that can modulate autophagy in specific contexts. It's a reminder that autophagy, like most biological processes, is neither purely good nor purely bad. It's a tool, and its effect depends on the context in which it operates. The connection between autophagy and longevity has been observed across an enormous range of species, from yeast to worms to flies to mice. In nearly every organism studied, interventions that extend lifespan also enhance autophagy, and genetic manipulations that enhance autophagy often extend lifespan. This correlation is so consistent that autophagy is now considered one of the key mechanisms underlying the effects of caloric restriction, the most reliable way to extend lifespan in laboratory animals. When you reduce food intake, you activate autophagy, and this activation appears to be necessary for at least some of the longevity benefits. But longevity is not just about living longer. It's about living well longer, maintaining health and function into old age. And here too, autophagy shows its importance. Animals with enhanced autophagy don't just live longer. They remain more active, maintain better cognitive function, and show delayed onset of age-related diseases. They experience what researchers call an extended health span, a longer period of healthy life. This is the real promise of understanding and supporting autophagy. Not necessarily adding years to life, but adding life to years, maintaining the vitality and capacity that make life worth living. The relationship between autophagy and exercise extends beyond the immediate effects we discussed earlier. Regular exercise, performed over weeks and months, induces adaptations in autophagy machinery. making cells more capable of mounting an autophagic response when needed. It's a form of training, conditioning your cells to be more efficient at cleanup and renewal. This may be one mechanism behind the protective effects of regular physical activity against a wide range of diseases. Your muscles remember the exercise you do, not just in their size and strength, but in the molecular machinery they maintain, including the components of autophagy. Different types of exercise may affect autophagy in different ways. Endurance exercise, like jogging or cycling, tends to promote autophagy, particularly in muscle and heart tissue, helping these tissues adapt to the demands of sustained activity. Resistance exercise, like weightlifting, also activates autophagy, but may do so in a pattern that's more focused on remodeling and growth, clearing away damaged components to make room for new muscle proteins. High-intensity interval training, which alternates between intense effort and recovery, may create particularly strong autophagy signals due to the rapid shifts in energy state. But all forms of movement, even gentle walking, provide some stimulus for cellular renewal. The phenomenon of exercise-induced autophagy may also explain some of the cognitive benefits of physical activity. When you exercise, autophagy increases not just in your muscles, but also in your brain, clearing away accumulated proteins and damaged mitochondria that might otherwise impair neuronal function. This may be one reason why regular exercise is associated with better cognitive function in older adults and reduced risk of dementia. Your brain benefits from the movement of your body, a reminder of the deep integration of all your systems. Autophagy also plays a role in the adaptive immune system, the part of immunity that learns and remembers specific pathogens. T cells and B cells, the key players in adaptive immunity, undergo dramatic changes in their autophagy activity as they respond to infection. Naive immune cells, those that haven't yet encountered their specific antigen, maintain relatively low autophagy. But when they're activated by encountering their target, autophagy increases to provide energy and building blocks for rapid proliferation. Then, as the infection is cleared and most of these cells die off, the few that survive to become memory cells adjust their autophagy to a level that supports their long-term survival. These memory cells can persist for decades, ready to respond quickly if the same pathogen returns, and autophagy is part of what allows them to endure. Vaccination works by creating these memory cells without requiring actual infection, and autophagy is involved in this process too. When you receive a vaccine, the antigens it contains are taken up by antigen-presenting cells, which process them and display fragments on their surface. This processing involves autophagy, which breaks down the antigens and helps load the fragments onto the display molecules. The quality of this autophagy-dependent processing can affect how well the immune system responds to the vaccine, how strong and long-lasting the immunity becomes. It's yet another example of autophagy working behind the scenes, supporting processes you might never have connected to cellular recycling. The role of autophagy in the endocrine system, the network of hormone-producing glands that regulates so much of your physiology, is equally important. Cells in the pancreas that produce insulin rely on autophagy to manage the stress of synthesizing and secreting this essential hormone. When these cells are young and healthy, autophagy helps them maintain the specialized structures needed for insulin production. As they age or face metabolic stress, autophagy becomes even more critical, removing damaged components and helping the cells survive. Impaired autophagy in pancreatic beta cells is associated with the development of diabetes, while enhancing autophagy can protect these cells and improve metabolic control. The thyroid gland, which produces hormones that regulate metabolism throughout your body, also depends on autophagy. Thyroid cells must produce large amounts of thyroid hormone and store it in an extracellular compartment called the follicle, then retrieve it when the body needs it. This complex process of synthesis, secretion, storage, and retrieval involves multiple forms of autophagy, helping cells manage the metabolic demands and maintain quality control. Disruptions in thyroid autophagy have been linked to various thyroid disorders, suggesting that supporting this process might help maintain thyroid health. Even the adrenal glands, which sit atop your kidneys and produce stress hormones like cortisol and adrenaline, utilize autophagy to manage their function. These glands must respond rapidly to stress, ramping up hormone production within minutes, and then return to baseline when the stress passes. Autophagy helps adrenal cells cope with the metabolic demands of this rapid response and also helps them recover afterward, clearing away the cellular stress that accumulates during periods of high activity. It's a system designed for resilience, for surging and recovering, across the countless small stresses of daily life. The relationship between autophagy and the extracellular matrix, the network of proteins and sugars that surrounds and supports cells and tissues, reveals another dimension of cellular recycling. Cells can use autophagy to degrade internalized pieces of extracellular matrix, breaking them down and either reusing the components or releasing them in modified form. This process helps cells remodel their surrounding environment, which is crucial during development, wound healing, and cancer progression. The extracellular space is not just a passive scaffold. It's an active participant in tissue function, and autophagy is one of the ways cells communicate with and reshape this space. Fibroblasts, the cells that produce most of the extracellular matrix in connective tissues, are particularly active in this regard. They continuously secrete matrix proteins and also continuously internalize and degrade them, maintaining a dynamic equilibrium that allows tissues to adapt to changing mechanical demands. When you stretch a tissue repeatedly, fibroblasts sense this and adjust their matrix production and degradation accordingly, strengthening the tissue in the direction of stress. Autophagy is involved in managing the cellular stress of this high secretory activity and also in degrading internalized matrix components. In the lungs, where delicate air sacs must remain thin enough for efficient gas exchange while strong enough to withstand the mechanical forces of breathing, autophagy helps maintain this critical balance. Lung epithelial cells use autophagy to manage the oxidative stress from constant exposure to oxygen and to clear away surfactant, the lipid protein mixture that reduces surface tension in the air sacs. Alveolar macrophages, immune cells that patrol the air sacs, rely heavily on autophagy to process the particles and pathogens they engulf. Impaired lung autophagy is associated with various lung diseases, from chronic obstructive pulmonary disease to pulmonary fibrosis, suggesting that this process is essential for maintaining the lung's delicate architecture. The heart, that tireless muscle that beats more than 2 billion times in an average lifetime, depends profoundly on autophagy to maintain its function. Cardiac muscle cells, or cardiomyocytes, are packed with mitochondria to meet their enormous energy demands, and these mitochondria must be continuously refreshed through mitophagy. The heart also faces unique mechanical stresses, constantly contracting and relaxing, and autophagy helps cardiac cells manage the protein damage that accumulates from this repetitive mechanical work. In heart failure, autophagy is often disrupted, and restoring it has shown promise in experimental models for improving cardiac function. The phenomenon of ischemic preconditioning, where brief periods of reduced blood flow can protect the heart against subsequent, more severe ischemia, appears to involve autophagy as one of its mechanisms. The initial brief stress activates autophagy, which clears away vulnerable cellular components and puts the cell into a more resilient state. When the more severe stress arrives, the cell is better prepared to survive it. This discovery has led to research into pharmacological approaches to mimic preconditioning, potentially offering protection against heart attacks and strokes by activating autophagy at the right time. In blood vessels, the endothelial cells that line the interior surface use autophagy to maintain the smooth, non-stick surface that allows blood to flow freely. These cells are constantly exposed to shear stress from flowing blood, and they use autophagy to manage this mechanical stress and to remove damaged components that might otherwise trigger inflammation or clotting. Endothelial autophagy is sensitive to flow patterns. Areas of smooth, laminar flow maintain healthy autophagy, while areas of disturbed flow, such as at branch points and arteries, often show impaired autophagy. This may be one reason why atherosclerosis, the buildup of plaques in arteries, tends to occur at these branch points. The connection between autophagy and cholesterol metabolism adds another layer to cardiovascular health. Cells can use autophagy to break down lipid droplets containing cholesterol esters, releasing free cholesterol that can then be exported from the cell or used for membrane synthesis. This process, sometimes called lipophagy when it focuses on lipid droplets, helps prevent the excessive accumulation of cholesterol in cells, including in the macrophages that infiltrate artery walls and contribute to atherosclerosis. Supporting autophagy may help these cells avoid becoming foam cells, the cholesterol-laden cells that are a hallmark of arterial plaques. Autophagy even extends its influence to the sensory systems that connect you to the world. In the eye, retinal cells use autophagy to manage the enormous metabolic demands of vision. Photoreceptors, the cells that detect light, are among the most metabolically active cells in the body, and they continuously shed and renew their outer segments, the part that contains the light-detecting machinery. This renewal process involves autophagy, which helps break down the shed outer segments that are engulfed by the retinal pigment epithelium, a layer of cells that supports the photoreceptors. Impaired autophagy in the retina is associated with age-related macular degeneration, a leading cause of blindness in older adults. In the ear, hair cells that detect sound vibrations and head movements also rely on autophagy to maintain their function. These cells are irreplaceable in mammals. Once lost, they don't regenerate, so maintaining them across a lifetime is crucial for preserving hearing and balance. Autophagy helps hair cells manage the mechanical stress of their function and the oxidative stress from their high metabolic activity. Supporting autophagy in the inner ear may be one approach to preventing or slowing age-related hearing loss, that gradual decline in auditory function that affects most people as they grow older. The sense of smell, mediated by olfactory neurons in the nose, provides another example. These neurons are unusual in that they do regenerate throughout life, being replaced every few months. But while they're alive, they depend on autophagy to maintain the specialized cilia that extend into the nasal cavity and detect odor molecules. The olfactory bulb in the brain, where these neurons send their signals, also shows high autophagy activity, particularly in the inner neurons that process and refine olfactory information. The gradual decline in sense of smell that many people experience with aging may be partly due to declining autophagy in this system. Even taste, that most direct and intimate of senses, involves autophagy. Taste receptor cells in the tongue are constantly exposed to the chemical environment of food and drink, and they use autophagy to manage this exposure and to maintain their receptors. These cells also have a relatively short lifespan, being replaced every week or two, and autophagy likely plays a role in the differentiation of new taste cells from stem cells in the taste buds. The connection between taste and metabolism runs deep. Taste receptors exist not just in the tongue, but also in the gut and other organs, where they help sense nutrients and regulate digestive processes. Autophagy in these systems may help coordinate the body's response to food, from the first taste to the final absorption of nutrients. The relationship between autophagy and body temperature regulation reveals yet another dimension of this process. When you're exposed to cold, brown adipose tissue, a specialized fat tissue that generates heat, increases its autophagy activity. This autophagy helps remodel the cells to increase their density of mitochondria, the organelles that produce the heat. It's a form of adaptation, preparing the tissue to be more effective at warming you. Conversely, when autophagy is impaired, the ability to adapt to cold is reduced. This may be one reason why maintaining autophagy function is important for metabolic flexibility, the ability to adapt to different environmental and nutritional conditions. The process of fever, that ancient response to infection where body temperature rises to help fight pathogens, also involves autophagy. The elevated temperature itself can trigger autophagy in many cell types, and this autophagy may contribute to the beneficial effects of fever by helping cells clear away damaged components and by enhancing the immune response. It's a reminder that many of the body's responses to stress, even uncomfortable ones like fever, are actually sophisticated adaptive mechanisms that promote survival and healing. Autophagy is woven throughout these responses, a common thread in the body's toolkit for managing challenges. As you lie here, perhaps feeling the weight of your body against the surface beneath you, perhaps noticing the quiet rhythm of your breathing, all of these processes are occurring within you. Your cells are recycling, renewing, maintaining themselves with a patience and precision that requires nothing from your conscious mind. The autophagy happening in your neurons, your heart cells, your gut lining, your immune cells, all of it proceeds according to ancient patterns, responding to signals both internal and external, maintaining the extraordinary organized complexity that is your living body. And within the deep complexity of cellular recycling, there exists a beautiful relationship with the simplest acts of living. When you breathe deeply, when you move gently through your day, when you rest in darkness, you are participating in an ancient conversation between your conscious choices and your cells' invisible wisdom. The autophagy that unfolds in response to a walk in morning light, the lysosomal activity that increases during your night's sleep, the proteasomal degradation that responds to the nutrients you consume. All of these are dialogues between the life you live and the life that lives within you. This is not control, but collaboration. Your cells do not wait for permission, yet they respond with exquisite sensitivity to the rhythms you establish, the stresses you encounter, the rest you allow. In the tissues of your reproductive system, cellular recycling maintains the delicate balance required for creation itself. The ovaries and testes, organs of potential, rely on autophagy to maintain the health of germ cells across decades. In the ovaries, autophagy helps preserve the finite pool of oocytes that a person carries from before birth, protecting these cells through years of metabolic stress, hormonal fluctuations, and time's passage. The oocytes themselves engage autophagy during maturation, clearing damaged mitochondria and misfolded proteins to ensure that if fertilization occurs, the resulting embryo begins with the cleanest possible cellular inheritance. This is recycling in service of the future, a molecular investment in generations not yet conceived. In the testes, autophagy supports the continuous production of sperm cells, a process that unfolds throughout adult life. The Sertoli cells that nurture developing sperm rely on autophagy to manage the enormous metabolic demands of spermatogenesis, recycling components to provide energy and building blocks for millions of new cells each day. Autophagy also protects developing sperm from oxidative damage, ensuring the integrity of the genetic material they will carry. Even the Leydig cells that produce testosterone engage autophagy to maintain their function across decades. The reproductive system, with its unique temporal demands, preserving some cells for years, producing others by the millions, demonstrates autophagy's remarkable flexibility, its ability to serve both conservation and abundance. And in the earliest moments of a new life, autophagy plays a role so fundamental that without it, development cannot proceed. Immediately after fertilization, the newly formed embryo faces a metabolic crisis. For several days, before it implants in the uterine wall and establishes a connection to maternal nutrients, the embryo must survive on the resources contained within the oocyte and the minimal nutrients present in the fallopian tube. During this critical window, autophagy becomes the embryo's primary source of energy and building blocks. The embryo digests its own cytoplasmic contents, recycling maternal proteins and organelles to fuel the rapid cell divisions that transform a single cell into a blastocyst. This is not damage or degradation, but transformation, the molecular substance of potential parents becoming the substance of a new individual. As the embryo develops, autophagy continues to shape its formation. During the process of gastrulation, when the simple ball of cells reorganizes into the 3 germ layers that will become all organs and tissues, autophagy helps orchestrate the massive cellular rearrangements required. Cells that must migrate, change shape, or differentiate into new types rely on autophagy to remodel their internal architecture, breaking down the proteins and structures of their former identity to make way for their new fate. The heart that begins to beat, the neural tube that folds and closes, the limb buds that extend. All of these developmental miracles involve countless cells recycling their contents in precisely timed waves, coordinated by signaling pathways that link autophagy to the master regulators of embryonic patterning. Even in the formation of fingers and toes, autophagy plays a gentle but essential role. The webbing that initially connects the digits must be removed for individual fingers to emerge, a process that involves programmed cell death. But before those cells die, they engage autophagy, recycling their contents one final time. The nutrients and energy recovered are not wasted, but are taken up by neighboring cells, contributing to the growth of the emerging digits. This is economy elevated to art. Even in dying, cells give back, their substance becoming part of the structures that will remain. The hand that will one day reach, grasp, create, begins its formation through this quiet molecular generosity. In the placenta, that temporary organ that exists solely to support the developing life, autophagy maintains the health of trophoblast cells that form the interface between maternal and fetal circulation. These cells face extraordinary demands, mediating the exchange of nutrients, gases, and waste products while also producing hormones that maintain pregnancy. Autophagy helps trophoblasts manage oxidative stress, recycle damaged mitochondria, and adapt to the changing oxygen levels they experience as the placenta develops. When autophagy is impaired in the placenta, pregnancy complications can arise: preeclampsia, intrauterine growth restriction, conditions that reflect the breakdown of the careful cellular maintenance that normally proceeds invisibly. The health of pregnancy rests, in part, on the quiet, continuous recycling happening in this temporary organ. And throughout childhood, as the body grows and develops, autophagy balances the competing demands of expansion and quality control. Growing tissues need abundant protein synthesis to build new structures, yet they also need autophagy to ensure that what is built correctly, that damaged or misfolded proteins are removed before they accumulate. In the developing brain, autophagy helps prune unnecessary synapses, refining neural circuits through a process that is as much about selective removal as about growth. The child's brain that learns language, recognizes faces, coordinates movement, is shaped not only by what is added, but by what is carefully, specifically taken away. This is growth through subtraction, complexity through selective simplification. In the immune system of the developing child, autophagy helps establish the repertoire of immune cells that will provide protection throughout life. As T cells and B cells mature, They undergo selection processes that eliminate cells with dangerous self-reactivities while preserving those that can recognize pathogens. Autophagy supports the survival of the cells that pass selection, helping them manage the metabolic stress of rapid proliferation and differentiation. The immune system that will protect against countless infections, that will remember vaccines and past exposures, that will distinguish self from other with remarkable precision, is built in part through autophagy's supportive presence during these critical developmental windows. As childhood gives way to adolescence, autophagy continues to support the dramatic changes of puberty. The surges of growth hormone, sex hormones, and other endocrine signals that drive adolescent development all influence autophagy in various tissues. In muscles that grow rapidly, autophagy helps balance protein synthesis with quality control, ensuring that the new muscle fibers are functional and healthy. In bones that lengthen and strengthen, autophagy in osteoblasts and osteoclasts helps coordinate the remodeling required for growth. Even in the brain, which undergoes significant reorganization during adolescence, autophagy contributes to the pruning and refinement of neural circuits, particularly in the prefrontal cortex, that region responsible for planning, impulse control, and complex reasoning. The adolescent brain, often described as a work in progress, is being shaped in part by the selective recycling of synaptic components, a molecular editing process that continues into the mid-20s. And in the transition to adulthood, autophagy shifts from supporting growth to maintaining stability. The body that has reached its full size now faces a different challenge: preserving function across decades, maintaining the integrity of cells that must last a lifetime. Autophagy becomes primarily a maintenance process, a continuous housekeeping that prevents the gradual accumulation of damage. This is the long middle of life, the years when the body's recycling systems work quietly to preserve the structures built during development. The heart that beats steadily, the lungs that breathe without thought, the kidneys that filter, the liver that detoxifies, all rely on autophagy to maintain their cellular populations in good working order. In the lens of the eye, a structure unique in the body, cellular recycling takes on a special character. Lens fiber cells, which must remain transparent to allow light to pass through, undergo a remarkable transformation during their maturation. They degrade their own nuclei and organelles, eliminating structures that would scatter light and interfere with vision. This is an extreme form of cellular recycling, a one-time event that creates cells that are essentially bags of crystalline proteins, perfectly arranged for optical clarity. Once this process is complete, these cells cannot engage in further autophagy. They have no machinery left to do so. They persist for the entire lifespan, their proteins never replaced, gradually accumulating damage that eventually leads to cataracts in old age. The lens demonstrates both the power of cellular recycling, creating perfect transparency, and its limits, the inability to maintain the perfection indefinitely without ongoing renewal. In contrast, the retina behind the lens engages in continuous, active autophagy throughout life. The photoreceptor cells of the retina, rods and cones, face an extraordinary challenge. They contain outer segments packed with the light-sensitive proteins and membranes required for vision. And these outer segments are constantly damaged by light exposure and the chemical reactions of phototransduction. To maintain function, photoreceptors continuously grow new outer segment material at the base while shedding old, damaged material from the tip. The shed tips are engulfed by the retinal pigment epithelium, a layer of cells behind the photoreceptors, which then uses autophagy and lysosomal degradation to break down and recycle the components. Each photoreceptor sheds about 10% of its outer segment every day, a remarkable rate of renewal. Your vision, your ability to read these words, to see the faces of those you love, to navigate the world, depends on this continuous cycle of growth, shedding, and recycling, a process so rapid and so essential that when it fails, blindness follows. The retinal pigment epithelium, those cells that perform this continuous recycling, are themselves long-lived cells that must be maintained across a lifetime. They rely on their own autophagy to manage the enormous load of photoreceptor material they digest each day. When autophagy in these cells becomes impaired with age, or due to genetic mutations, the result can be age-related macular degeneration, a leading cause of vision loss in older adults. The disease reflects the accumulation of undegraded material, the formation of deposits called drusen, and eventually the death of photoreceptors and the cells that support them. It is a reminder that recycling is not optional, not a luxury, but a fundamental requirement for sustained function. The eyes that see are eyes that recycle continuously, tirelessly, across decades. In the inner ear, where sound waves are converted into neural signals, cellular recycling supports the maintenance of hair cells, the sensory receptors of hearing. These cells, topped with delicate stereocilia that bend in response to sound vibrations, face continuous mechanical stress. Autophagy helps hair cells manage this stress, recycling damaged proteins and organelles to maintain their function. In mammals, hair cells are not replaced if lost. The ones you were born with must last your lifetime, making their maintenance through autophagy particularly critical. Age-related hearing loss, a nearly universal experience in humans, reflects in part the gradual decline of autophagy and other quality control mechanisms in these cells, the slow accumulation of damage that eventually impairs their function. The sounds that fade with age, the highest frequencies, the subtle consonants, mark the limits of cellular maintenance, the eventual exhaustion of recycling capacity. Yet even as some systems decline, others maintain remarkable stability. The heart, that tireless muscle, continues to beat with extraordinary reliability across decades, in large part because of effective cellular recycling. Cardiomyocytes, the muscle cells of the heart, are mostly postmitotic. They do not divide and replace themselves in any significant number after early childhood. The cells that make up your heart today are largely the same cells you had as a child, now decades older, yet still contracting rhythmically, still pumping blood. They achieve this longevity through intensive autophagy, continuously recycling their abundant mitochondria, turning over their contractile proteins, removing damaged components before they can interfere with function. The heart is a testament to the power of maintenance, a demonstration that with sufficient recycling, cells can function reliably for a human lifetime. When the heart is stressed by high blood pressure, by ischemia, by disease, autophagy often increases as a protective response. The heart cells ramp up their recycling to cope with the increased demand, to manage the damage caused by reduced oxygen or increased workload. This is adaptation, the cellular equivalent of tightening the belt, conserving resources, prioritizing survival. Sometimes this adaptation is sufficient, and the heart recovers, its cells restored through enhanced recycling. Sometimes the stress is too great, too prolonged, and even increased autophagy cannot prevent damage. But the attempt is always made. The cells always engage their recycling machinery in response to threat, a biological optimism encoded in ancient pathways. In the vascular system, the network of blood vessels that carries blood to every tissue, autophagy maintains the health of endothelial cells, the single cell layer that lines all vessels. These cells are directly exposed to the sheer stress of flowing blood, to fluctuations in oxygen and nutrients, to inflammatory signals. Autophagy helps endothelial cells manage these stresses, maintaining their barrier function, their ability to regulate vascular tone, their role in preventing clot formation. When endothelial autophagy is impaired, the result is endothelial dysfunction, a condition that underlies atherosclerosis, hypertension, and many cardiovascular diseases. The health of your circulation, the ease with which blood flows to nourish your tissues, depends on the continuous recycling happening in the cells that line your vessels, a maintenance process that normally proceeds without your awareness, without your effort. In the smooth muscle cells of vessel walls, autophagy plays a more complex role. These cells can proliferate and migrate in response to vessel injury, contributing to the repair of damaged vessels, but also, when dysregulated, to the formation of atherosclerotic plaques. Autophagy in smooth muscle cells can be protective, removing damaged organelles and preventing cell death. But excessive autophagy can also contribute to plaque instability. The balance is delicate, context-dependent, a reminder that cellular recycling is not always simply beneficial, but must be appropriately regulated. The body navigates these complexities through intricate signaling networks, adjusting autophagy levels in response to local conditions, attempting to optimize outcomes in the face of competing demands. In the blood itself, cellular recycling takes a different form. Red blood cells, which lack nuclei and most organelles, cannot engage in autophagy. They have no machinery for it. Instead, they rely on other quality control mechanisms, particularly the proteasome, to maintain their hemoglobin and other essential proteins during their roughly 120-day lifespan. When red blood cells become too old, too damaged, they are recognized and removed by macrophages in the spleen and liver, which then recycle their components. The iron from hemoglobin is carefully recovered and returned to the bone marrow for incorporation into new red blood cells. The amino acids from degraded proteins are released into circulation for use throughout the body. This is organismal-level recycling, the body recovering and reusing the valuable components of worn-out cells, a process so efficient that almost no iron is lost. The iron in your blood today has likely been recycled through countless generations of red blood cells. White blood cells, in contrast, are nucleated cells fully capable of autophagy, and they use it extensively. Neutrophils, the most abundant white blood cells, are short-lived cells that engage autophagy both for their normal function and during their death. When neutrophils respond to infection, swarming to sites of bacterial invasion, they use autophagy to help manage the intense metabolic demands of their antimicrobial activities. They also use a specialized form of cell death called enetoses, in which they expel their DNA and associated proteins to form neutrophil extracellular traps that can ensnare bacteria. Autophagy proteins participate in this process, helping to orchestrate the controlled release of cellular contents. Even in dying, neutrophils use their recycling machinery, repurposing it for a final defensive act. Lymphocytes, the T cells and B cells of adaptive immunity, use autophagy throughout their lifespans. Naive lymphocytes, circulating in the blood and lymph awaiting their first encounter with antigen, maintain themselves in a quiescent state partly through basal autophagy, which provides the minimal energy and building blocks needed for survival without proliferation. When a lymphocyte encounters its specific antigen and becomes activated, it rapidly downregulates autophagy and upregulates protein synthesis, shifting its metabolism to support rapid proliferation and differentiation. Then, after the infection is cleared and most of the activated lymphocytes die off, the few that survive to become memory cells re-engage autophagy, using it to support their long-term survival. Memory T cells can persist for decades, ready to respond rapidly if the same pathogen is encountered again, and they maintain themselves during this long wait partly through autophagy, recycling their components to sustain their slow, steady metabolism. In the bone marrow, where blood cells are continuously generated throughout life, autophagy supports the hematopoietic stem cells that give rise to all blood cell types. These stem cells face a particular challenge. They must persist for a lifetime, dividing occasionally to produce more stem cells and the progenitor cells that will differentiate into mature blood cells, all while maintaining their own integrity and avoiding the accumulation of damage. Autophagy is essential for this long-term maintenance, helping hematopoietic stem cells remain quiescent, manage metabolic stress, and preserve their regenerative capacity across decades. When autophagy is impaired in these stem cells, their function declines, leading to reduced blood cell production and increased risk of leukemia. The continuous renewal of your blood, the replacement of millions of cells every second, ultimately depends on the health of a small population of stem cells maintained in part through cellular recycling. In the thymus, that small organ behind your breastbone where T cells mature, autophagy plays a crucial role in T cell development. As T cells develop, they undergo positive and negative selection, processes that test their T cell receptors and eliminate cells that are either non-functional or dangerously self-reactive. The thymic epithelial cells that mediate this selection rely on autophagy to present a diverse array of self-antigens, including proteins from all tissues of the body. Through a process that involves autophagy, these epithelial cells display fragments of cellular proteins on their surface, allowing developing T cells to test their receptors against a comprehensive library of self. T cells that react too strongly to these self-antigens are eliminated, preventing autoimmunity. This is recycling in service of education, the breakdown of cellular contents creating the curriculum that teaches immune cells to distinguish self from other. The thymus itself undergoes a remarkable change with age. It gradually involutes, shrinking and being replaced by fatty tissue, a process that begins in adolescence and continues throughout life. This involution is associated with declining T cell production and is thought to contribute to the weakening of immune function with age. Interestingly, autophagy in thymic epithelial cells declines during involution, and experimental enhancement of autophagy can partially restore thymic function in aged animals. This suggests that the age-related decline of the thymus, long thought to be inevitable, might in fact reflect in part the declining capacity for cellular recycling, a potentially modifiable aspect of aging. The possibility that enhancing autophagy might rejuvenate the aging thymus, restore T cell production, and strengthen immune function in older adults is an area of active research, a hope grounded in the fundamental biology of cellular maintenance. In the lymph nodes, those small bean-shaped organs distributed throughout the body, autophagy supports the function of the diverse cell types that orchestrate immune responses. Dendritic cells, which capture antigens from tissues and present them to T cells and lymph nodes, use autophagy both to degrade captured material and to generate the peptide fragments displayed on MHC molecules. B cells and lymph nodes, when activated by antigen and T cell help, undergo rapid proliferation and differentiation into antibody-secreting plasma cells, a process that involves dramatic cellular remodeling supported by autophagy. Even the stromal cells that provide the structural framework of lymph nodes engage autophagy to maintain their function. A lymph node responding to infection is a hub of cellular activity, with millions of cells dividing, differentiating, and interacting. And throughout this activity, autophagy provides essential support, managing metabolic stress, enabling cellular remodeling, contributing to antigen presentation. In the spleen, that fist-sized organ in your upper left abdomen, autophagy serves multiple functions. The spleen filters blood, removing old red blood cells, capturing bloodborne pathogens, and mounting immune responses. The macrophages that engulf aged red blood cells rely on lysosomal degradation to break down the cellular contents and recover iron. The lymphocytes in the spleen's white pulp use autophagy in ways similar to those in lymph nodes. The spleen also serves as a reservoir of immune cells, particularly monocytes, that can be rapidly mobilized in response to injury or infection, and autophagy helps maintain these cells in their quiescent state until they are needed. The spleen, often called a forgotten organ because people can survive without it, is in fact a sophisticated recycling and immune hub, continuously filtering your blood, recovering valuable components, and maintaining immune readiness. In the liver, that largest of internal organs, autophagy reaches perhaps its highest expression. Liver cells, hepatocytes, are metabolic powerhouses responsible for processing nutrients, synthesizing proteins, detoxifying harmful substances, and countless other functions. They are also extraordinarily rich in autophagy. Under normal fed conditions, hepatocytes maintain a basal level of autophagy, continuously recycling their abundant organelles. During fasting, hepatic autophagy increases dramatically, becoming a primary source of amino acids for gluconeogenesis, the production of glucose to maintain blood sugar levels. The liver, in a sense, digests itself to feed the rest of the body, a molecular altruism that sustains life during times of scarcity. This hepatic autophagy is not random degradation, but is carefully regulated to preserve essential functions while providing needed resources. Certain proteins and organelles are preferentially spared, while others are rapidly recycled. The liver maintains its ability to perform its essential functions even while engaging in extensive self-digestion, a testament to the precision of autophagic selectivity. When you break a fast, when you eat and insulin levels rise, hepatic autophagy is suppressed, and the liver shifts to anabolic metabolism, synthesizing and storing nutrients. This rhythmic oscillation between autophagy and anabolism, between breakdown and building, is part of the liver's normal metabolic cycle, a daily tide of cellular recycling that responds to your eating patterns. The liver's remarkable regenerative capacity also involves autophagy. If part of the liver is removed, the remaining tissue rapidly proliferates to restore the organ to its original size, a regenerative feat almost unique among human organs. During this regeneration, autophagy is dynamically regulated, initially increasing to provide resources for the energy-intensive process of cell division, then decreasing as the new tissue is built. The regenerating liver is a site of intense cellular activity, with autophagy and anabolism working in coordination, recycling providing the raw materials that synthesis transforms into new tissue. This regenerative capacity, this ability to rebuild itself, makes the liver remarkably resilient, able to recover from injury, infection, and even significant surgical resection. Yet the liver's recycling capacity is not infinite. Chronic liver diseases, hepatitis, fatty liver disease, cirrhosis, often involve dysregulation of autophagy. In fatty liver disease, impaired autophagy contributes to the accumulation of lipid droplets in hepatocytes, the defining feature of the condition. The cells lose their ability to effectively recycle stored fat, leading to progressive accumulation, cellular dysfunction, and eventually inflammation and fibrosis. In cirrhosis, the end stage of chronic liver disease, extensive scarring replaces functional tissue, and autophagy is profoundly disrupted. The liver that once recycled so efficiently becomes a site of failed maintenance, accumulated damage, and lost function. Yet, even in disease, the liver cells continue to attempt autophagy, continue to engage their recycling machinery, a persistence that sometimes allows for recovery if the underlying cause is addressed. In the kidneys, those paired organs that filter your blood and regulate fluid balance, autophagy maintains the health of the highly specialized cells that perform these essential functions. The proximal tubule cells, which reabsorb the vast majority of filtered nutrients and water, are packed with mitochondria to power the energy-intensive transport processes they perform. These cells rely heavily on mitophagy to maintain their abundant mitochondrial populations, continuously recycling damaged mitochondria and replacing them with healthy ones. When autophagy is impaired in proximal tubule cells, kidney function declines, and susceptibility to acute kidney injury increases. The kidneys that filter your blood, that maintain your body's fluid and electrolyte balance, that remove waste products, depend on the continuous recycling happening in their tubular cells. In the glomeruli, the tiny filtering units of the kidney, autophagy maintains the specialized cells called podocytes that form the filtration barrier. Podocytes are remarkable cells with long foot processes that interdigitate to create a selective barrier, allowing small molecules to pass while retaining proteins and blood cells. These cells are postmitotic. They cannot divide and replace themselves, so they must be maintained for a lifetime. Autophagy is essential for this maintenance, helping podocytes manage metabolic stress, recycle damaged organelles, and preserve their complex architecture. When podocyte autophagy is impaired, the result is often proteinuria, the leakage of protein into urine, a sign of failing kidney filtration. Many kidney diseases involve podocyte injury, and autophagy enhancement has shown promise in experimental models as a way to protect these critical cells. In the intestines, that long tube of digestion and absorption, autophagy serves multiple essential functions. The intestinal epithelium, the single cell layer that lines the gut, is one of the most rapidly renewing tissues in the body, completely replacing itself every few days. This rapid turnover involves stem cells at the base of intestinal crypts continuously dividing to produce new epithelial cells, which then migrate up the villus, mature, perform their absorptive functions, and are eventually shed into the intestinal lumen. Throughout this journey, autophagy supports these cells, helping them manage the metabolic demands of their rapid proliferation and the stresses they encounter in the harsh intestinal environment, fluctuating nutrients, microbial products, digestive enzymes. Paneth cells, specialized epithelial cells at the base of intestinal crypts, use autophagy in a particularly interesting way. These cells produce antimicrobial peptides that help control the intestinal microbiome, preventing bacterial overgrowth and maintaining the balance of microbial communities. Autophagy in paneth cells is required for the proper packaging and secretion of these antimicrobial peptides. When autophagy is impaired in paneth cells, their function is disrupted, the intestinal microbiome becomes dysregulated, and susceptibility to intestinal inflammation increases. Certain genetic variants that impair autophagy are associated with increased risk of Crohn's disease and inflammatory bowel disease, highlighting the connection between cellular recycling and intestinal health. Goblet cells, another specialized intestinal cell type, produce the mucus that coats the intestinal lining, providing a protective barrier between the epithelium and the intestinal contents. These cells synthesize enormous amounts of mucin proteins, which must be properly folded and assembled before secretion. The endoplasmic reticulum of goblet cells faces a tremendous protein folding burden, and autophagy, particularly autophagy, helps these cells manage this burden, removing misfolded proteins and excess membrane. The mucus layer that protects your intestines, that allows food to pass smoothly, that prevents bacteria from directly contacting your cells, is produced by cells that rely heavily on cellular recycling to manage their specialized function. In the intestinal immune system, which contains more immune cells than any other organ, autophagy plays crucial roles. The gut-associated lymphoid tissue, Peyer's patches, isolated lymphoid follicles, mesenteric lymph nodes, contains vast numbers of T cells, B cells, and other immune cells that must distinguish between harmless food antigens and commensal bacteria on one hand, and dangerous pathogens on the other. This discrimination requires education, and autophagy contributes to this education by helping present antigens to developing immune cells. The intestinal macrophages and dendritic cells that sample antigens from the gut lumen use autophagy to process these antigens and present them in ways that promote tolerance to food and commensals while maintaining responsiveness to pathogens. The relationship between autophagy and the gut microbiome extends beyond immune regulation. The bacteria that inhabit your intestines produce metabolites, short-chain fatty acids, vitamins, signaling molecules that are absorbed by intestinal cells and influence their function. Some of these microbial metabolites affect autophagy in intestinal cells, either promoting or inhibiting it depending on the specific metabolite and context. The short-chain fatty acid butyrate, for example, produced by bacterial fermentation of dietary fiber, has been shown to influence autophagy in colonocytes, the epithelial cells of the colon. This creates a feedback loop. Your diet influences your microbiome. Your microbiome produces metabolites. These metabolites influence autophagy in your intestinal cells, and autophagy in turn influences intestinal health and immune function, which affects the microbiome. You are not recycling alone. You are recycling in conversation with trillions of microbial partners. In the pancreas, autophagy maintains the health of both the exocrine cells that produce digestive enzymes and the endocrine cells that produce hormones. The acinar cells of the exocrine pancreas synthesize and secrete enormous amounts of digestive enzymes, proteases, lipases, amylases, that are released into the small intestine to break down food. These cells face a unique danger. The enzymes they produce are capable of digesting the cells themselves if prematurely activated. Autophagy helps protect acinar cells, removing damaged organelles and misfolded proteins that might trigger inappropriate enzyme activation. When autophagy is impaired in acinar cells, they become more susceptible to pancreatitis, a painful and potentially dangerous inflammation of the pancreas. The beta cells of the pancreatic islets, which produce insulin, rely on autophagy to maintain their function across decades. These cells must sense blood glucose levels and secrete precisely calibrated amounts of insulin in response, a task that requires healthy mitochondria for glucose sensing and abundant ER for insulin synthesis. Autophagy, particularly mitophagy and phagy, helps beta cells maintain these organelles in good working order. In type 2 diabetes, beta cell function gradually declines, and impaired autophagy may contribute to this decline. The beta cells accumulate damaged mitochondria, misfolded insulin, and other cellular debris, leading to cellular dysfunction and eventually cell death. The loss of beta cell mass and function in diabetes reflects, in part, failed cellular maintenance, the gradual exhaustion of recycling capacity under conditions of chronic metabolic stress. Interestingly, the alpha cells of pancreatic islets, which produce glucagon, appear to be more resilient than beta cells, and this resilience may relate to their more robust autophagy. Alpha cells maintain better autophagic function with age and metabolic stress, which may explain why they are less susceptible to the dysfunction seen in diabetes. This difference between cell types within the same microenvironment illustrates how cellular recycling capacity can vary and how this variation influences susceptibility to disease. The hope is that understanding these differences might reveal ways to enhance beta cell autophagy, potentially preserving their function and preventing or slowing the progression of diabetes. In adipose tissue, the body's fat stores, autophagy plays complex and context-dependent roles. Adipocytes, the cells that store fat, can engage in lipophagy, a selective form of autophagy that targets lipid droplets for degradation. During fasting or exercise, when the body needs to mobilize stored fat for energy, lipophagy contributes to the breakdown of triglycerides, working alongside other lipolytic pathways. Autophagy in adipocytes also influences their health and function. Impaired autophagy is associated with adipocyte dysfunction, inflammation, and insulin resistance. The fat tissue that stores energy, that cushions organs, that produces hormones influencing appetite and metabolism, is maintained in part through cellular recycling. In brown adipose tissue, a specialized fat that burns calories to generate heat, autophagy plays a particularly important role. Brown adipocytes are packed with mitochondria, which contain the protein UCP1 that allows them to generate heat by uncoupling respiration from ADP production. These abundant mitochondria must be maintained through mitophagy, and brown adipocytes have high rates of mitochondrial turnover. When you are exposed to cold, brown adipose tissue is activated, increasing its metabolic activity and heat production, and this activation is associated with increased autophagy. The thermogenesis that keeps you warm, that burns calories, that contributes to metabolic health, involves cellular recycling working in coordination with mitochondrial respiration. The discovery that adult humans retain functional brown adipose tissue, once thought to be present only in infants, has sparked interest in its potential role in metabolic health. People with more active brown adipose tissue tend to be leaner and have better metabolic profiles, and interventions that activate brown fat, cold exposure, certain hormones, exercise, show promise for improving metabolism. Autophagy appears to be part of this story, supporting brown adipocyte function and potentially contributing to the metabolic benefits of brown fat activation. The possibility that enhancing autophagy might increase brown fat activity, boost calorie burning, and improve metabolic health is an intriguing area of ongoing research. In muscle tissue, both skeletal and cardiac, autophagy must be carefully balanced. Too little autophagy and damaged components accumulate, impairing muscle function. Too much autophagy and muscle mass is lost, leading to atrophy. Muscle cells navigate this balance through sophisticated regulation, adjusting autophagy levels in response to nutritional status, exercise, hormones, and aging. After resistance exercise, when muscle cells are building new protein to increase strength and size, autophagy is generally suppressed to allow net protein accumulation. But even during this anabolic phase, some basal autophagy continues, removing damaged proteins and organelles to ensure that what is built is functional. The muscle that grows stronger, that adapts to training, does so through a carefully orchestrated interplay of synthesis and recycling. In aging muscle, autophagy often declines, contributing to sarcopenia, the loss of muscle mass and strength with age. Aged muscle accumulates damaged mitochondria, protein aggregates, and other cellular debris, reflecting impaired autophagy and other quality control mechanisms. This accumulation impairs muscle function, reduces regenerative capacity, and increases susceptibility to injury. Exercise, particularly resistance training, can partially counteract these changes, enhancing autophagy in aged muscle and helping to maintain its health. The elderly person who maintains strength and mobility through regular exercise is, at the cellular level, maintaining autophagy, keeping their muscle cells recycling systems active, preventing the accumulation of damage that would otherwise occur. The satellite cells that enable muscle regeneration also rely on autophagy. These muscle stem cells, residing in a quiescent state along muscle fibers, can be activated in response to injury to proliferate and differentiate into new muscle fibers. Autophagy supports satellite cells during their quiescence, helping them survive in their low metabolic state, and during their activation, helping them manage the metabolic demands of proliferation. In aged muscle, satellite cell function declines, and impaired autophagy may contribute to this decline. The reduced regenerative capacity of aged muscle, the slower recovery from injury, reflects in part the aging of stem cells and their recycling machinery. In tendons and ligaments, the connective tissues that attach muscles to bones and bones to each other, Autophagy maintains the health of fibroblasts, the cells that produce and maintain the collagen-rich extracellular matrix. These tissues are relatively acellular. Most of their volume is extracellular matrix, but the cells they contain must be maintained across a lifetime. Tendon fibroblasts engage in autophagy to manage the metabolic demands of collagen synthesis and to respond to mechanical loading. When you exercise, the mechanical stress on tendons activates, signaling pathways that influence autophagy, contributing to tendon adaptation and strengthening. The tendons that transmit force, that enable movement, that withstand enormous loads, are maintained by cells that recycle their components in response to the stresses they experience. In cartilage, the smooth tissue that cushions joints, autophagy is essential for maintaining chondrocyte, the cells embedded in the cartilage matrix. Cartilage is avascular. It has no blood supply, so chondrocyte exist in a low oxygen, low nutrient environment. They rely heavily on autophagy to survive in these harsh conditions, recycling their own components to generate energy and building blocks. Autophagy in chondrocyte declines with age, and this decline contributes to osteoarthritis, the degenerative joint disease that affects millions. The cartilage that wears away in arthritic joints reflects, in part, the death of chondrocyte that can no longer maintain themselves through recycling. Interventions that enhance autophagy in chondrocyte have shown promise in experimental models of osteoarthritis, suggesting that supporting cellular recycling might slow or prevent joint degeneration. In bone, autophagy maintains the health of osteocytes, the cells embedded in the mineralized bone matrix. Osteocytes are the most abundant cells in bone, and they serve as mechanosensors, detecting mechanical loading and orchestrating the bone remodeling response. Like chondrocyte, osteocytes exist in a challenging microenvironment with limited access to nutrients and oxygen. They rely on autophagy for long-term survival, and when autophagy is impaired in osteocytes, bone quality declines. The bones that support your weight, that protect your organs, that serve as a reservoir of minerals, are living tissues maintained by cells that continuously recycle their contents. The osteoblasts that build new bone and the osteoclasts that resorb old bone also engage autophagy. Osteoblasts, during their active phase of bone formation, use autophagy to manage the metabolic demands of synthesizing and secreting enormous amounts of collagen and other bone matrix proteins. Osteoclasts, the large multinucleated cells that dissolve bone mineral and degrade bone matrix, use autophagy to support their unique function. The continuous remodeling of bone, the removal of old or damaged bone and its replacement with new bone, involves cellular recycling at every step. Your skeleton is not a static structure, but a dynamic tissue, constantly being recycled and renewed, maintained through the coordinated activities of multiple cell types, all engaging their autophagic machinery. In the skin, that largest of organs, autophagy supports the health of keratinocytes, the cells that form the protective outer barrier. As keratinocytes mature and migrate from the basal layer to the surface, they undergo a specialized form of differentiation that involves extensive autophagy. The cells gradually degrade their nuclei and organelles, eventually becoming corneocytes, the flattened, dead cells of the stratum corneum that provide the skin's barrier function. This is programmed cellular recycling, a developmental process that creates a protective barrier through selective self-destruction. The skin that protects you from the environment, that prevents water loss, that keeps out pathogens, is formed through autophagy. In the dermis beneath the epidermis, fibroblasts produce and maintain the collagen and elastin that give skin its strength and elasticity. These fibroblasts engage in autophagy to manage the demands of extracellular matrix production and to respond to stresses such as UV radiation. Sun exposure damages skin cells, generating oxidative stress and DNA damage, and autophagy helps manage this damage, removing damaged organelles and proteins. However, chronic sun exposure can overwhelm recycling capacity, leading to the accumulation of damage that manifests as photoaging, wrinkles, loss of elasticity, uneven pigmentation. The skin that ages, that shows the marks of sun exposure and time, reflects in part the limits of cellular recycling, the gradual accumulation of damage that exceeds the capacity for repair. In hair follicles, autophagy supports the rapid cell proliferation required for hair growth. Hair follicles cycle through phases of growth, regression, and rest, and during the growth phase, cells in the hair bulb divide rapidly to produce the hair shaft. Autophagy provides the energy and building blocks for this rapid proliferation. When autophagy is impaired in hair follicles, hair growth is affected and susceptibility to hair loss increases. The hair that grows from your scalp, that you cut and style, is produced by cells engaged in intensive recycling to support their rapid division. In melanocytes, the cells that produce the pigment melanin, autophagy plays a role in managing the oxidative stress associated with melanin synthesis. The production of melanin generates reactive oxygen species as a byproduct, and melanocytes must manage this oxidative stress to avoid damage. Autophagy helps remove oxidized proteins and lipids, protecting melanocytes from the toxic effects of their own metabolic activity. The pigmentation of your skin and hair, the colors that vary so beautifully across human populations, is produced by cells that rely on recycling to manage the stresses of their specialized function. In the nervous system, autophagy's importance reaches perhaps its peak. Neurons, those long-lived cells that cannot be replaced, depend utterly on effective recycling to maintain their function across a lifetime. The accumulation of damaged proteins and organelles in neurons is a hallmark of neurodegenerative diseases, Alzheimer's, Parkinson's, Huntington's, ALS, and reflects the failure of autophagy and related quality control mechanisms. The brain that thinks, remembers, feels, and controls the body requires continuous cellular recycling in its billions of neurons. When that recycling fails, cognition declines, movement becomes impaired, and eventually the person is lost, even as the body remains. Yet, even in the face of neurodegenerative disease, the brain cells continue to attempt autophagy, continue to engage their recycling machinery. This persistence offers hope because it suggests that enhancing autophagy, supporting the brain's own maintenance mechanisms, might slow or prevent neurodegeneration. Numerous experimental approaches are being explored. Drugs that activate autophagy, gene therapies that enhance the expression of autophagy genes, lifestyle interventions like exercise and fasting that naturally boost autophagy. The goal is to support the brain's recycling, to help neurons manage the protein aggregates and damaged organelles that threaten their function, to extend the years of healthy cognition. And as you rest now, as your mind perhaps drifts towards sleep, autophagy is unfolding in every cell of your body. In your neurons, synaptic proteins are being recycled, memories are being consolidated, cellular housekeeping is being performed. In your muscles, damaged mitochondria are being removed, proteins are being turned over, preparations are being made for tomorrow's movements. In your liver, metabolic shifts are occurring, autophagy is increasing in response to your overnight fast. Amino acids are being generated for gluconeogenesis. In your immune cells, surveillance continues. Autophagy supports the long-lived cells that protect you, enables the presentation of antigens, contributes to the defense against any pathogens you encounter today. This is the gift of cellular recycling. It happens without your effort, without your awareness, a biological grace that sustains you. You do not need to think about it, to control it, to manage it. Your cells know what to do. They have been doing it for billions of years. perfecting these processes through endless generations, encoding them in genes that are expressed in every cell. The autophagy that unfolds in your body tonight is the same fundamental process that occurs in yeast, in worms, in flies, in every organism that has been studied. It is ancient, conserved, essential. It connects you to the deep history of life, to the first cells that discovered that survival requires not just building, but also breaking down, not just synthesis, but also recycling. And in this continuity, in this connection to all life, there is something deeply comforting. You are not separate, not isolated, but part of an unbroken chain of living systems that have maintained themselves through recycling for eons. The cells in your body are engaging in the same molecular processes that sustained your ancestors, that sustain all living things, that will sustain your descendants. This is biology as heritage, as gift, as ongoing story. You are a chapter in this story. Your life a brief expression of these ancient processes. Your cells recycling as cells have always recycled, maintaining themselves, maintaining you, with a fidelity and persistence that asks nothing in return. So rest now, knowing that you are cared for at the deepest level, that your cells are working on your behalf, that the recycling that sustains life is unfolding within you. You are safe in this knowledge, held by these processes, supported by this molecular maintenance. The autophagy that occurs in darkness, in silence, in the privacy of your cells, is a form of biological love, a commitment to your continued existence encoded in the very structure of life. You are recycled, renewed, maintained, not through your effort, but through the ancient wisdom of your cells. And in this, you can find peace, can let go, can trust that the work of living at the cellular level will continue while you rest. You are held. You are maintained. You are, in the deepest sense, cared for. And that is enough.